দেখাচ্ছি meeting is now streaming live on YouTube.
Now it's doing.
হ্যাঁ, বাকিদের এবারে ইয়ে করে অ্যাকসেপ্ট করে নেবে তো?
হ্যাঁ, বাকি এলে অ্যাকসেপ্ট করুন। এই নেটটা দেখা যাচ্ছে না, দেখ তো ওয়াইফাই।
Sir, now I admit all.
ওয়াইফাইটা একটু দেখে দিতে।
Sir, please শুরু করে দিন।
Am I audible?
Yes.
Good morning, everybody.
Morning.
We are here for a seminar organized by Department of Botany, Vivekananda Mahavidyalaya, Hooghly. The topic is international webinar on COVID-19 treatment and understanding of human immune response, a complex circuit for vaccine research. The first session is the inaugural session.
এখানে আসছে না।
First session is inaugural session, and the welcome address will be provided by Dr. Ashim Kumar Samanto. He is our honorable principal. May I request sir to please deliver your welcome address.
Okay. Thank you, Dr. Sina. Very good morning to everybody. Good morning to our honorable speakers, Dr. Bhaumik, Dr. Rahman, and Dr. Rai Chodori. Welcome and congratulations to our President, Honorable President of the governing body, Sri Bablu Gayan, other members of the honorable members of the governing body, our IQAC coordinator, Dr. Chatterjee, and all other participants from India as well as from the abroad. It's a great pleasure. It's a great pleasure to us that the Department of Botany, rather Dr. Sangram Sinha and his colleagues, in collaboration with the IQAC coordinator, is going to organize.
Excuse me, sir.
Okay.
Please put on your video.
Okay, thank you.
Okay. Now okay. Please carry on.
It's a great pleasure to us that the Department of Botany, rather Dr. Sangram Sinha, the HOD of the department, and other colleagues, in collaboration with the IQAC coordinator, is going to organize such an international webinar. The topic is very relevant today's. The topic is COVID-19 treatment and understanding of human immune response related with the vaccine research also. I congratulate to our honorable speakers who has contributed their valuable times for this purpose for the day. It is well known to us that the global people are facing a great challenge. Whether we are in great trouble due to COVID-19 or the SARS-CoV-2 infection. We know that according to the comment of the director who we are not just fighting an epidemic, we are fighting an infodemic also. Actually, COVID-19 has developed a medical emergency and a global crisis so rapidly that it first emerged in December 2019, and on 11th March 2020, it was declared as a pandemic by the WHO. Whereas by the 6th April 2020, globally, there are more than 1.2 million confirmed cases of COVID-19 detected, and about 67,000 deaths occurred across the more than 200 countries, as per WHO update. But on 1st August, the figure reached about 1.75 crore in respect of global aspect, the infected people, where deaths occurred about 6.78 lakhs. In our country, in Indian respect, the figure already reached 17 lakhs infected by the COVID-19. And the deaths were more or less 35,000. So it's a great panic to us. The infection not only restricted in the individual or their family members, it also spread to the healthcare system, food system, and also economics. It is well known to us that the common symptoms include mainly fever and cough, but a proportion of patients may develop shortness of breath. And in more severe cases, the infection can cause pandemic, that is pneumonia, acute respiratory distress syndrome, organ failure, and death. Perhaps, unfortunately, you know that there has been a lot of misinformation and myths that have spread like wildfire through social media and other channels, which cause a great confusion to us. However, due to COVID-19, our lives have been turned upside down. Rather, COVID-19 discloses our physical, physiological, clinical, as well as mental weakness. Rather, it is exposed that we are how much unorganized in respect to the such a teeny object, which neither living nor a non-living material. Hopefully, many scientists, healthcare professionals are working out at the global level to understand the virological, epidemiological, and clinical aspect of COVID-19 in order to identify the effective drugs and vaccines. One of the recent developments regarding the understanding of SARS-CoV-2 infection in the mechanism of immune response, which involved during the pathogenesis, the cytokine storm syndrome during infection in the patient, against which a drug has been designated various drugs have been designated. I hope today's discussion may focus on various aspects of a novel corona infection, such as global epidemiology, genome organization, immunopathogenesis, transmission cycle, treatment and control strategies also. Thanks to our honorable speakers, they will highlighted all the aspects related to the COVID-19. Again, I thanks to all the participants from India as well as from the abroad. Thanks a lot.
Thank you very much, sir, for your very relevant and interesting welcome address. Now, I would like to request Dr. Partho Chatterjee, IQAC coordinator and head Department of Physics, Vivekananda Mahavidyalaya, Hooghly, to kindly address the audience with your keynote. May I request Dr. Chatterjee, please.
Excuse me, sir. Please unmute yourself.
Thank you, Dr. Sangamchila, for your introduction. On behalf of the Internal Quality Assurance Cell, Vivekananda Mahavidyalaya, Hooghly, I welcome you all to this early morning session of this webinar on COVID-19. We have on board two eminent speakers, Dr. Habibur Rahman and Dr. Siddhartha Bhomik. They will enlighten us on the various aspects that is on this COVID-19 situation. Our principal, Dr. Ashwin Kumar Samanto, has already elaborated the pandemic situation that is actually ravishing our whole universe right now. We are all in a very pandemic situation. We are all we are living in a fearful environment. We are fearful in going out of our house. We are fearful about our fellow human beings, fellow citizens, fellow friends. Personally, I'm also in a fearful situation because just a few days back, my brother and his entire family, they're living in Ahmedabad. They caught COVID-19. My brother's wife and his daughter, 13 years daughter, they caught COVID. Fortunately, they came out of the situation. They are now all free from this. But another unfortunate event happened in my family also. My cousin brother, who was suffering from cancer, died a few days back, most probably of this infection. I feel very sorry for the people who have died all over the world. So at present, we are in a very difficult situation. We do not know whether a vaccine will ever come out to the market and help us in this very gory moment. But we can hope for the best. We can hope that someday, maybe at the end of this year or early next year, we will have one vaccine in the market, and probably we will come out of this pandemic situation. One of the things that pains me right now is we are following this social distancing norm to control this pandemic. This is taking toll on our lives. It's taking toll on our fellow human being. It's taking toll on our fellow human feeling. We are somehow losing our human fellow feeling. We should all work together to bring back our human fellow feeling and hope that someday we'll come out of this pandemic situation. The world will be very beautiful as it was. Thank you very much.
Thank you, Dr. Partha Chatterjee. In spite of your mishap in your family, and you are still, I feel you are still in a condition where you need some time to stabilize yourself, but in spite of your problems, you extended your hand to help us. Thank you very much for your keynote address. I pay my sincere gratitude to you. Thank you very much. Now, I'm glad to have with us Sri Bablu Gayan, President, Governing Body. Now, I would like to call upon Dr. Sri Bablu Gayan, President, Governing Body. Sir, please deliver your special address to the audience. Bablu Gayan, sir, please.
নমস্কার, যারা যারা আছেন প্রত্যেককে শুভেচ্ছা এবং অভিনন্দন জানাচ্ছি। সমগ্র বিশ্বে অদৃশ্য করোনা ভাইরাস কোভিড-19 আতঙ্কে মানুষ জর্জরিত। বিশ্বে কোটি কোটি মানুষ এই মারণ রোগে আক্রান্ত হচ্ছেন এবং প্রায় 6,71,000 মানুষ ইতিমধ্যেই এই রোগে প্রাণ হারিয়েছেন। আমাদের দেশ ও রাজ্যেও আক্রান্তের সংখ্যা ক্রমশ বৃদ্ধি পাচ্ছে। করোনা যোদ্ধা যারা আছেন, ডাক্তার, নার্স, স্বাস্থ্যকর্মী, আমাদের পুলিশ প্রশাসন, সরকার এর বিরুদ্ধে লড়াই সংগ্রাম চালিয়ে যাচ্ছেন। সারা বিশ্বের বৈজ্ঞানিকগণ টিকা আবিষ্কার করার জন্য আপ্রাণ চেষ্টা চালিয়ে যাচ্ছেন। পাশাপাশি কোভিড-19 চিকিৎসা পদ্ধতি এবং প্রতিরোধ বিষয় নিয়ে হরিপাল বিবেকানন্দ মহাবিদ্যালয়ের বটানি বিভাগ, বিশেষ করে এইচওডি ডক্টর সংগ্রাম সিনহা এবং তার ডিপার্টমেন্টের অন্যান্য অধ্যাপক অধ্যাপিকাদের নিয়ে এই সেমিনার যে আয়োজন করেছেন, আলোচনা সভা, তার জন্য ওনাদেরকে আমি আন্তরিকভাবে ধন্যবাদ জানাচ্ছি। ইতিমধ্যেই এই সেমিনারে বক্তব্য রেখেছেন আমাদের হরিপাল বিবেকানন্দ মহাবিদ্যালয়ের মাননীয় অধ্যক্ষ ডক্টর অসীম কুমার সামন্ত মহাশয়, এবং বক্তব্য রাখলেন আমাদের IQAC কোঅর্ডিনেটর ডক্টর পার্থ চ্যাটার্জি মহাশয়। আজকের এই সেমিনারে যারা অতিথিগণ আছেন, ডক্টর সিদ্ধার্থ ভৌমিক, ডক্টর মোহাম্মদ হবিবুর রহমান, ডক্টর কৌশিক রায়চৌধুরী সহ আরো অন্যান্য যারা যারা আজকের এই সেমিনারে আছেন, প্রত্যেককেই আমি আন্তরিকভাবে ধন্যবাদ এবং কৃতজ্ঞতা জানাই। এবং আমাদের হরিপাল বিবেকানন্দ মহাবিদ্যালয়ের গভর্নিং বডির অন্যান্য মেম্বার যারা সকলেই এই সেমিনারে উপস্থিত আছেন ডক্টর করবী মান্না, ডক্টর অভিজিৎ পাকিরা, শ্রী অলোক সাঁতরা, শ্রী স্বরূপ মিত্র, শ্রী প্রদীপ দলুই, গভর্নিং বডির মেম্বার আমাদের চন্দনবাবু আছেন, সঞ্চয়িতা ম্যাডাম আছেন আমাদের অশিক্ষক কর্মচারী বন্ধুদের পক্ষ থেকে শঙ্কর শুভ্র কোলে সহ সবাই যারা আছেন প্রত্যেককেই আমি ধন্যবাদ জানাই। এবং আজকের এই মিটিং যে আপনারা অ্যারেঞ্জমেন্ট করেছেন এই করোনার ভাইরাস নিয়ে, আজকে সর্বত্রই এই যে চেষ্টা, এই চেষ্টাকে সাধুবাদ জানাই। এর আগেও আমাদের কম্পিউটার সায়েন্স ডিপার্টমেন্ট এবং ইংলিশ ডিপার্টমেন্ট হরিপাল কলেজের আমাদের তন্ময় বাবু বা আমাদের সঞ্চয়িতা ম্যাডাম ওনাদের উদ্যোগেও এই ধরনের একটা সেমিনার হয়েছিল। আজও যে এই সভা হচ্ছে আলোচনা সভা, এই আলোচনা সভা সেমিনার সর্বাত্মক সুন্দর হোক, সাফল্যমণ্ডিত হোক, এই কামনা করে সকলকে শুভেচ্ছা, ভালোবাসা, নমস্কার জানিয়ে আমি এখন বক্তব্য শেষ করি।
Thank you very much, President sir, for your nice address to the audience. Here we close the closing session of closing of the inaugural session. We will move on to the technical session. In our technical session, we'll have 3 speakers. First one, Dr. Siddhartha Bhumik. Second one is Dr. Habibur Rahman, and third one is Dr. Kawasaki Syndrome. Dr. Khan, Dr. Sunil Khan, are you audible? Am I audible to you, Dr. Khan? Devbroto, please unmute Dr. Khan.
He is unmuted from our end.
Hello, Devbroto.
Sir, he is unmuted from our end. Please wait, sir.
Okay. Okay, we'll move on to the technical session. Here I'd like to introduce my distinguished speaker, Dr. Siddhartha Bhumik. I pay my sincere gratitude to Dr. Bhumik for donating his valuable time. The topic is very relevant in the sense that more than 190 countries in the world is affected. Everybody is looking for a solution, but still there is no solid solution has come out. In this present context, the topic is very relevant, and all the schools, colleges, and universities are now being closed. The only platform is to share, is to disseminate our knowledge through this webinar. Many colleges and universities are organizing webinars. We are also not far behind. Here we have our speaker, Dr. Siddhartha Bhumik. He did his PhD from Department of Infectious Disease and Immunology from IICB. After completion of B.Pharm and M.Tech from Jadavpur University, then he joined as a postdoctoral fellow in the University of Washington. Then he worked on the different viruses like flavivirus, West Nile virus. In the year 2011, He joined the Emroy Vaccine Center, Emroy, Atlanta. Then there he promoted as a research associate in Emroy University Vaccine Center and worked on the dengue and Zika virus. Now he is holding the senior scientist position and analytical project leader, analytical science and technology, emergent biosolutions in Baltimore. He is presently engaged in developing COVID-19 vaccine and anthrax monoclonal antibody therapeutic drugs. He has published more than 20 papers in peer-reviewed journals. He has patent on his name, soluble protein antigen as vaccine and therapeutic drug against Leishmania. He has got a very big CV to his name, and within this limited time, maybe I'm not justifying to his credit, but I've tried to highlight his achievements. Now, may I request Dr. Bhomik to kindly deliver his talk. Dr. Bhomik, please.
All right, so hope you all can see my screen, right?
Yes, yes.
Yeah,
Yes,
yes, I can see.
Okay, all right. First of all, I would like to thank all the organization committee members of Vivekananda Mahavidyalaya and especially the Mr. Principal of Vivekananda Mahavidyalaya and the IQAC coordinator and the president of the governing body and especially Sangram Shina for organizing this nice seminar. And as everybody has said, this is a really tough time for not just India, but the entire world, who everybody is suffering from COVID-19. And the topic that I have chose is very relevant right now. It's the recent advancements that have happened for the COVID-19 vaccine research. And I am extremely involved in this field right now with the company that I'm working with. We are developing 4 vaccine candidates, and that is a disclosure that I want to make. So all the 4 vaccine candidates that we are making are the lead candidates right now throughout the world. So I would not be able to answer any questions which says to choose what is the best vaccine candidate which we might be able to get in the market soon. So, but I will highlight all the positives and the negatives and all the differences between all the vaccine candidates that are being right now being tried. All right, with that, I will I'll start with the introduction of the COVID-19. I'll talk a little bit about the viral structures, the disease prevention, which is the main focus of my topic on discussing what the vaccine candidates that there are. And then I'll end up with, if you have any questions around what I have discussed in this presentation. So first to introduce, before even I can go over and start my introduction on COVID-19 or coronavirus 2019, there has been a history of epidemics which the world has seen. I think this is one of the largest epidemics that we have seen in our lifetime. So that is where we are feeling that this is something which is of major importance to us. But if you look at the specifics of the epidemics and pandemics that have been in this world, this is one of the very smallest pandemics that have had hit our world, starting from the bubonic plague around in 1300, where more than 200 million people have died, to the smallpox pandemic in the early 15th, 16th century, where about 56 million people died. And to the recent, people have been comparing the recent pandemic of COVID-19 with the Spanish flu, which happened around 1912, where about 50 to 60 million people who have died. And still ongoing, the HIV epidemic, where about 35 million people have died. But most of these pandemics have happened long time back, and though none of them have been registered much in the history, and we have not really experienced this hands-on. And that's the reason why we feel that the COVID-19 current pandemic is something which has changed our lives. But if you look at the size of these balls, these are comparing the number of people who have died. So if you look at the size of these balls, the COVID-19 is really small in compared to that. And here is another picture of the size of the people who have died for different pandemics. And you can see that COVID-19 is one of the smallest pandemics that have ever hit the world. But the real problem which we definitely feel is before there wasn't a whole lot of intercommunication, and the world was not as small as it is right now. So that's the risk. So the communication and the information about people getting sick, people dying, are reaching to every person. And that's for the reasons why there is a whole lot of panic around this COVID-19. So just to give you a brief history about, or a brief introduction about COVID or coronavirus, it's the name comes from the crown because it has a spike. If you look at the electrogram picture that is provided at the background, you see these small spikes around the viral structure, from which this name comes as coronavirus, and it's a family of Coronaviridae. There has been there are 4 different subtypes of coronavirus, primarily the alpha, beta, gamma, and delta, depending upon the genomic structure. And alpha and beta coronavirus are the ones which mainly infect the mammals and causes respiratory infection, and it also infects animals, which causes gastroenteritis. So as everybody is pretty much aware that there is a animal host that where coronavirus harbors and lives, and sometimes it jumps onto humans, and that is where one of this coronavirus also had the start. And also there is a gamma and delta coronavirus, which is generally found in geese, guinea pigs, pigeons, ducks, parrots. So all these animals and birds which are around us, they also carry the other strain of coronavirus, the gamma and the delta coronaviruses. So we are already like around our environment, we have animals which have coronaviruses, but it is very rarely that these coronaviruses would jump into humans and cause this pandemic which we see right now. So this is not the first coronavirus outbreak that has happened. Before this December 2019 COVID-19, there has been 6 different coronaviruses that have plagued the world, and which mainly there had been small, mild cold symptoms, and that is the reason there has been not a whole lot of importance given to COVID-19 coronaviruses, other than the last 2 coronaviruses before this, which people are really aware of, is the 2003 SARS epidemic, where about 10% of the people died who had coronavirus. And in 2012, there was a similar coronavirus, which is called MERS, because it originated in Middle Eastern Asia, and where the mortality was even higher, where about 37% of the people have died. But if you look at the 2019, which started from Hooghly in a province in China, where this is a novel beta coronavirus, and right now, if you look at the mortality rates, it's about 4 to 5%. So which if you look crunch numbers, it's not really a whole lot of people who have been dying, but it's a lot of people getting infected. And if you look at the viral structure of this current coronavirus, or the COVID-19, it is like any other RNA virus, where the structure is very simple, where it has this blue spike proteins, and with that there is a envelope which covers the genetic material, which is called the envelope protein, and then there is a small red like structures which are the membrane protein which hold the structure together, and there is the nucleocapsid or N protein which actually holds the genetic material inside the virus, and which is used actually to transmit or transfer the genetic material into the infecting cells. And if everybody, I am sure, are now aware of this, that this spike protein or these blue structures, these are the ones where everybody is targeting for making the vaccine, because this structure is the one which actually the COVID-19 virus latches onto the ACE2 receptor of the epithelial cells, which actually is the main target right now for binding of the virus to the epithelial cells of the lungs mainly. But this ACE2 protein actually, or receptor, is also found in intestines, kidneys, and other blood vessels, where this spike protein binds to this receptor, and that is how it is internalized into into the epithelial cells. And this ACE2 expression of the ACE2 is hugely increased in patients who have type 1 or type 2 diabetes, as well as who have hypertension, because of these people taking ACE2 inhibitors to reduce their diabetes or their hypertension. And this causes the increase in the expression of the ACE2, which also therefore does increase the chance of having the person getting infected more. And that is one of the reasons why we see people who have diabetes and who have high blood sugar are most likely to have been infected with COVID. And there has been also some studies right now ongoing where people with hypertension are the drug to reduce the hypertensions, which are generally the ACE2 inhibitors. People are trying to provide some other types of blood pressure reducing drugs, so that the chances of higher expression of the ACE2 is diminished. But as I said, the spike protein is the main protein which is being used to target the cells, and that is the main protein which all the vaccine peoples are trying to use to target, so that it can stop the virus from infecting. So this gives me a right segue to go into the preventions of the disease. And as I said, my main topic of discussion today would be around the prevention and not the treatment. So I will just be focusing on the disease prevention. As everybody knows, treatment is good, but I think prevention is better. So everybody has heard that prevention is better than cure. So if we don't want to get infected, it's better to prevent that infection from happening, and then rather than getting infected and trying to figure out a drug to treat and reduce that infection. So there are a few ways of prevention. The first is the most easiest, and that is what people are doing right now, is social distancing and wearing masks. But this cannot go on forever, because we're living in a society where we meet with people, and until we have a safe vaccine or a drug, this is the best way to prevent the disease from spreading. But as I mentioned, that this cannot go in forever. We have to travel, we have to go out, we have to do our business. So we cannot just hold on and live a life where we have to distance from each other, as well as we have to wear masks throughout our entire life. This cannot happen. So this is a stopgap measure till we have a vaccine or a drug for this COVID-19. The second way of prevention is natural infection, which everybody is aware of, and people have been talking about herd immunity, which I'm sure with all this news going around, so people have been talking about herd immunity, where a particular set of people gets infected, about 60 to 70% of the people, and which will then help the other people who have not been infected to be protected, because most of the people in the community has been infected, have been exposed to the virus. But natural infection has helped previously in previous diseases, especially like cholera, chickenpox, measles, polio, where if somebody gets infected or somebody get, say, in our life, we have also seen if you get infected with chickenpox, you would not have chickenpox for your rest of your life. There are exceptions if you have low immune cells in your body, or if you have a condition, you might have a relapse of chickenpox or measles, but most likely if you have an infection, you will never have it. But do you want to go that route? That's the biggest question, because you might have chickenpox and it might go away, but it can also cause pneumonia, and there have been instances where people have died for chickenpox, or else for Haemophilus influenza, where people had Haemophilus influenza and it has given protection, but there have been people who have had intellectual disability. Also rubella, which can be cured, but having rubella, you might have people who might have birth defects due to rubella. So do we want to take that risk that we would have everybody get infected with COVID-19 and have build up a herd immunity? So that is a risk. First of all, because we don't know much about this virus. So we don't even know if the immune memory is long lasting, which means that if you have the virus once, will you ever be protected for your life, or if the protection will only be for a few months? If the immune memory is not long lasting, you might again have a chance of COVID-19. And as everybody knows that about 5% people definitely are getting succumbed to this disease. So we don't want to take that risk of having a herd immunity be the way of getting this virus eliminated from this world. So the most or the talked about prevention is the vaccination, and vaccination is not is very similar to natural infection. It is how we fake our immune system, or we trick our immune system to think that there has been a natural infection, and our immune cells, the T cells and the B cells, will act to generate cell-mediated immunity and humoral immunity against this virus, for which we'll have antibodies that are generated, which will latch onto the virus and inactivate the virus, or for T cells which will get activated and kill the cells that get infected with the virus. So this is a technique that we have learned from natural infection for our body to understand that there has been an infection happening. And this vaccination is not a new phenomenon. It has its roots a long time back. About in 11th century, there has been historical data that people have shown from China, that the vaccination or vaccination of a virus or a bacteria has happened, where people who have had infections, people take that infection and inoculate that another separate person with that in small dose of infection, and that is how the protection happens. Nowadays, this is no longer the route that people take. There have been lot of advances in vaccinations, because the chances of infecting people with a live virus or a bacteria, there is a high chance that the person might get also severely sick. So right now, there are about 5 different types of vaccinations that are available in the market. The oldest is, as I said, is a live attenuated vaccine, where the virus or the bacteria is still alive, but it has been attenuated, which means that the virus or the bacteria, there has been some mutations done, or some changes done to the gene so that it cannot or no longer are able to make the people very sick. There might be a small infection that might happen, but that would not create a huge infection, which or else the virus or the bacteria would have been able to do. So some of the examples are vaccinia, the smallpox vaccines, or MMR or rotavirus as zoster virus, which are live attenuated vaccines. There have been the next, the second type of very popular vaccine type is inactivated or killed vaccine, which is the safest. So this, or one of the safest, the others also very safe. So where the virus or the bacteria is being killed by heat inactivation or by formalin treatment, and that inactivated virus or the killed virus is then introduced into the healthy individuals to get protection. And the third type of vaccine is a toxoid vaccine, which is created from the toxoids, which are for bacteria. The toxins are the ones which are the dangerous part. So you make the toxins inactivated and make toxoids vaccine. And one of the very common vaccines is Tdap. Anybody who gets a cut or a bruise would get this Tdap vaccine. So this is a very common vaccine that are used. The recent advancement of the vaccines are the subunit vaccines, where instead of this whole inactivated or whole live attenuated vaccines, a small portion of the active ingredient or the seq protein of interest of the virus or the bacteria are actually inoculated, which will give a robust immune response. So first you have to determine which part of the virus or the bacteria will give a high or strong immune response. You take that small piece of that protein and make subunit vaccines. And hepatitis B, pneumococcal or human papillomavirus vaccines are examples of subunit vaccines. And lastly, the other type of vaccine that is also now coming up in the market is a conjugate vaccine. This is only provided when the antigen is not that strong immunogenic, which means that it doesn't provide a robust immune response if it has been given individually or injected individually to the human. So it is being tagged with another immunogen, which is a very strong immunogen, which will generate very strong and robust immune reaction. So once these two tagged antigens are injected into the human, so with the use of that strong immunogen, the weak immunogen also gets a huge boost. So that is how a conjugate vaccine is developed. And there are a couple of conjugate vaccines right now in the market, the Haemophilus influenza vaccine and the typhoid conjugate vaccine. This is a very important slide, and one must appreciate the amount of work that has gone through in this field of vaccinology, where if you look at this slide, let me point out the extreme left-hand side, the dates are when a disease has been identified. And if you see a vertical line, this is where actually the vaccine has come to the market. So this is the time difference that is present right now from when a disease has been identified to when a vaccine has been approved to be used in the market. So as you can see, there are a few diseases where there is no vertical line, which means the vaccine has not yet come to the market. So if you look at the influenza, it was first identified in early 1900, and the first influenza vaccine was developed in 2004. So it took about 60 years for the scientists to develop a vaccine. If you look at malaria, the first signs of malaria was identified in 1880, but still now we don't have an approved malaria vaccine. There has been a lot of vaccine candidates. I always try to stress upon vaccine candidates, because all around the world right now, there is a news that there are vaccines. So vaccine is something once you have a regulatory person or regulatory agency, be it the FDA for US or for India, the drug control for India, when it provides a stamp that now this is a vaccine that can be used for normal use or for general use, that is when a vaccine becomes effective or a vaccine comes to the market. Till then, any vaccines that have been tried around in individuals are actually vaccine candidates. And I will bring a slide later on where I will show you that about 94% of the vaccines which start off from the preclinical, there is only 4-6% of the vaccines which end up in the market. So even though there is a huge hype about a lot of vaccines, there will be only a handful which will actually be available in the market. So if you look at this graph, you'll see that for typhoid, meningitis, pertussis, which is whooping cough. So there has been slowly the time difference from when the disease has been identified to when the vaccine has come into the market, the time difference has started to go down from 60 years to 30, 40 years to chickenpox is about, again, 50 years to measles, only like about 20 years to cervical cancer, human papillomavirus, which was the latest vaccine that has been approved for commercial use. There has been other vaccines which have come to the market, but this is one which is globally used. There has been vaccines which are only given to like travelers or given to certain demographic people so that those people can be protected from some diseases. But the cervical cancer is the latest in the line of vaccines that have been approved, which was first identified in 1981, and in 25 years we see that in the market. So if you look at the whole time difference, so from a vaccine to be developed and approved from 80 years, we now have it in 25 years. And for COVID-19, what we are expecting is to have a vaccine in 1 year. So that is a huge accomplishment in the scientific community to have a vaccine, at least in the market, in 1 to 2 years, where this has never happened before. So this is something that one has to be very mindful of when we are talking about vaccines. So as I said, so the fastest vaccine that has been approved in the market has been 25 years. So everybody would have a question, like, why does it take so long for a vaccine to come to the market? I think the biggest reason being the biopharmaceutical companies, they are very reluctant to invest in R&D for production of a drug or a therapeutic which does not generate continuous revenue, right? So if you have a drug for a disease, you will always be having it. Say, for instance, if somebody is infected if somebody has hypertension, the person will be taking the hypertension drug forever till the person dies, right? But if you have a vaccine, the person will never be infected or will get sick with that disease. So the cost factor is very huge for a pharmaceutical industry to actually invest in vaccines. The next, to develop a vaccine and to bring it to the market, right now, it takes at least $1 billion for a vaccine to develop. And this is one of the least amounts. So there can be more than that. You can have $2 to $3 billion and still you can have a failed vaccine. So this is a huge commitment as far as the biopharmaceutical companies are involved. And there are extensive steps, which I'll go into details in my next couple of slides from a vaccine that is being developed in the lab to come to the market. And as I said in my previous slide, that about 94% of the vaccines that start off as vaccine candidate actually fail to come to the market. Only 6% of the vaccines actually which start off from a trial actually ends up in becoming an approved vaccines. So with that, I will start off with the different stages of vaccine development. The first and foremost is the candidate identification of what the candidate vaccine would be. It can be, as I said, there are different types of vaccine types. It can be a live attenuated vaccine. It can be subunit vaccine. So what type of right clone should be used? What type of sequence for the protein we need to identify. All of these things have first get identified, and that happens in the lab. And as everybody knows, all good things happen from small labs. So this is where everything begins. And then the preclinical studies, where generally the studies are done in animals, and most likely the animal model that are generally tested are the ones which are very similar to have the disease efficacy or disease symptoms to humans, so that when you provide or administer the vaccine candidate, you can have a chance that to observe if the disease progresses or what the outcome of it is, so that it can be replicated in humans. And next, the first human study that happens is the clinical phase 1 trial. The clinical phase 1 trial is the primary objective of the clinical phase 1 trial is to determine the dose, the safety, and of the vaccine candidate, to understand if there is any reaction that happens to the individual. Say if the vaccine is given and people start to have rashes or fever, or the part of the body gets paralyzed and stuff, like you cannot have the vaccine go to the next stage. So these are the main objectives in a phase 1 trial. So you have, everybody would have heard about a lot of phase 1 trials that have happened. So these are the only things that are determined for phase 1 trial. And that is also the true for the COVID-19 vaccine. People just look at doses, the safety, efficacy, and if there is any reactions. And there might be some secondary objectives to look at antibody responses and neutralizing antibody titers, but they do not look at the efficacy, which is the most important thing. So after phase 1 trial, even if any company does say that our vaccine is working really well, so that is something based on only on the antibody responses. And antibody responses does not mean that you will be protected. There can be antibody responses which can be just binding antibody, but which cannot neutralize the virus. So that way the vaccine will not be an effective vaccine. So there is a lot of news and media outlets that keep on telling people that the phase 1 is successful, which I would say that it is only a half story. From phase 1 to phase 3 and getting approval, there is a huge path to follow. And for the phase 1, the most important thing is it happens only in a very small number of people, and generally it happens in only a very specified cohort of people. I mean, so this vaccine will be given to only people who have been very well screened, only in a small subset of population who are very similar, not from different parts of the country, not from people who have different ethnic backgrounds and stuff like that. So it is a very regulated study that happens. The next, once a vaccine passes the phase 1 trial, they will move on to the phase 2 trial, where so the phase 1 would have around 200 people. Now you would have around few hundred to 1,000 people, depending upon the amount of money the company wants to spend for the phase 2 trial. And also, this is just also determining the safety. But most important thing that people look at the phase 2 trial is to identify the dose. So the phase 1, if you look very closely and actually read the study materials of the vaccine trials, you'll see that all the vaccine candidates, they try about 3 or 4 different types of doses. So in phase 2, the primary objective is to identify the optimal dose that they will be using if the vaccine is successful to move on to the phase 3. Because there is no more trial and error that can happen in phase 3. So people look at doses, people look at schedules. The scheduling means whether you have to give just 1 vaccine or you have to give 2 vaccines. Do you have a booster? How many days apart will the booster be given? And also in phase 2, they want to go to multiple sites. As I said, the phase 1 is generally at 1 site. Say the vaccine trial happens only in Kolkata. And not anywhere else. The phase 2, the vaccine trial will happen in Kolkata, Delhi, Mumbai, and if the company has money, it might happen Europe and Australia and US also. And it will also have a much more wider demographic people, as well as across different age of people, and also mixing different male versus female. So there is a much more diversity that is required. And this is very important because every individual has a different immune response. So that is also something that one has to look very carefully when designing a vaccine, an effective vaccine. And once we have a successful phase 2 trial, it moves on to phase 3. So this is a much more larger scale enrollment, where it can be a few thousand people to about 30 to 50,000 people, depending upon how severe the disease is. So if the incidence of the disease in the population it's a huge impact on depending on the size. If the incidence is very low, say right now for COVID, the incidence is very high. But if you are studying for a vaccine for a disease which is very low, say, for instance, my example of like working with leishmania, people hardly have leishmania anymore right now. So for that, to have a successful vaccine, you have to have a huge set of population, so that you know that the people who get infected versus the people who get protected. But for COVID right now, because we are on a stage of where a current epidemic is happening, with a low number of people, you can also determine if the vaccine is efficacious or not. So if the incidence definitely plays a role in determining the study population. The next, the trials are typically designated to evaluate efficacy. And this is the first time when actually we have to look at vaccine efficacy. What does efficacy means? Efficacy means whether the vaccine actually protects. So before this, all the studies that were done are on healthy individuals who have been tracked only to identify if the vaccine is safe, or if the vaccine has elicited antibody responses. It can be binding antibody, it can be neutralizing antibody, but generally neutralizing antibody still can dictate whether the vaccine will work or not. Binding antibody does not. Antibody that is generated in a human after the vaccination can bind to a virus or the bacteria, but it might not neutralize. So that is something one needs to be really careful when looking at the data that is coming out in the newspapers or in the news channels or in published articles. So binding antibody or antibody titers does not really mean that the vaccine actually works. So here right now, people will look at efficacy, which means that half of the people will be vaccinated, half of the people will not be vaccinated. The people who will not be vaccinated, there will be a certain percentage of the people who would get infected. So that will be compared to the people where the vaccination has happened, and then they will compare that. So if the vaccination would not happen, this many people would have been infected. So if the vaccination was able to protect, so how many people got protected? That is what the actual or the true data of a vaccine, if it is effective or not, that will come from phase 3 trials. And that is what, as I mentioned, the vaccine efficacy is defined by the percentage reduction in incidence among the vaccinated people. So, and this is not where it ends. So when the vaccine phase 3 trial, if it demonstrate efficacy and safety, all the primary and secondary endpoints, targets that the trial they have to previously disclose it, what their expectations are. If those are met, then the manufacturer will provide an application to the regulatory agency. As I said, for US, it is FDA. And once you get the license, then you start the manufacturing of the vaccine, and then you can sell it in the market. So this is a huge step, a lot of investment, a lot of money that is at stake, because vaccine trials are never easy. Vaccine trials are never cheap. People do not get vaccines for free. If something happens, there is a lot of money that needs to be provided to the individuals. So people are always really cautious. But right now for COVID-19, since it is a huge global pandemic or epidemic that's ongoing, a lot of the companies are taking the risk of doing all these studies, compiling into one, starting to manufacture the vaccines at risk. Even though it fails, they are ready to lose that money. Government is providing a lot of support for that, and that is the reason we are being able to shrink this timeline of like 4 or 5 years to 10 years of the entire process to about a year. And in the last segment of my talk, I will just provide the main COVID-19 vaccine candidates right now. So as you can see, there are about 170 vaccine candidates till a week back. Of that, 140 are still in preclinical, 19 are in phase 1, 11 are in phase 2, and 3 are in phase 3. They are not even in phase 3. None of the vaccines are yet in phase 3. There is all the enrollments are happening, and the phase 3 are starting next month for most of the vaccines. So as you can see, there is 170 vaccine candidates. They all have a common goal. And I'm sure not all the 170 vaccine candidates will reach the finish line. There will be a handful, but it is a good sign that there is so many companies who have invested the money. So at least a few companies can finally finish that end goal to have the vaccine that people can be administered when it becomes approved and comes to the market. So the target of all these vaccine candidates are to generate a robust antibody response that can neutralize the virus, as well as to generate a strong T-cell immune response that can actually destroy the infected cell. So as I said, I think only like a handful, maybe 10% of these 170 vaccine candidates will ultimately end up in the market. So, but since there are so many vaccine candidates that are being tried, the hope is very high that we will have a vaccine soon. So of that 170 vaccine candidates, I have listed a few vaccine candidates which I feel are way ahead in the game compared to the others. The first is the CanSino which is which has been developed in China. It is an adenovirus expressing the S protein. If you all recollect, the S protein is a spike protein where people are targeting this because this is the most immunogenic epitope, and this is where the vaccine actually binds to the ACE2 protein of the epithelial cells to get internalized. So that is where this vaccine candidate is. The AD5 is a adenovirus vector which is replication deficient. So that means it cannot replicate, but it can express or it can be translated to generate the COVID S protein, and against that S protein, the body can generate antibody responses. So this is right now going into phase 3 trial. I will discuss more about each of these vaccine candidates, the ones which are in phase 2, phase 3 right now, and tell about where they stand. The next is the Cyanobac. This also is developed in China. This is an inactivated whole virus. So the whole virus is killed, and then the whole virus is injected into the human body. The third is the AstraZeneca, which is developed in UK with the collaboration of Oxford University, and it has been developed and manufactured in US and different parts of the world. Also in India, this has been, the manufacturing has happened, but this has not yet reached phase 3. The phase 3 trial will begin next month. So this is also very similar to the CanSino biologics, where the adenovirus expressing S protein. The only difference between these two are this is a chimpanzee adenovirus, and the chimpanzee would not create an antibody response in humans, but what we are mostly concerned about is the S protein against which our body can generate antibody responses. The fourth is the Moderna. This is a US-based company which are making mRNA-based vaccines. And this is one of the two leading candidates of mRNA vaccines. The other is the Pfizer-BioNTech. The BioNTech is a German company where also are making mRNA-based vaccines. These are all moving into phase 3 trials soon. So these two, the only word of caution about these two vaccine candidates are that there has been no mRNA-based vaccine yet in the market. There has been instances where people have tried, but they have all failed. They have not been able to cross the phase 2. So these two vaccine candidates have at least crossed the phase 2 hurdle. The other one is the Inovio Pharmaceuticals, which is a DNA-based vaccine, which is now enrolling for phase 2. Codagenics. This is a US-based vaccine. This is a live attenuated whole virus. It is entering phase 1. And the last, but a very strong candidate is the Sanofi GSK vaccine. They are the last to join the race, but as I said, vaccine business leads a lot of money, and Sanofi and GSK both has that. And as Sanofi is a leader in vaccines throughout the world. So even though they have just completed phase 1, they are a strong candidate to have a robust vaccine soon. And their vaccine is very, very tried and tested. It's a protein-based vaccine. That is what most of the vaccines that we take are. So they are going by the most tried and trusted vaccine type. So there are very high hopes on Sanofi and GSK, even though they just started phase 1 right now. So with that, I will move on to each of the vaccine candidates that are moving into phase 3 next month. And I would just provide you where they stand right now. Starting off with CanSino Biologics, where the phase 2A study was published, where 508 participants were provided the vaccine at just one site in China. All these vaccines were given to, so this is the biggest advantage of this vaccine study yet with the published data, that all the adults that were given were over the age of 18. So most of the vaccine studies that you will see or hear in the paper or here in the news, they are all given between the age of 18 to 55. And as everybody knows, the COVID-19 is generally infecting or people getting sick more are the people over the age of 55. This vaccine was tried with people also over the age of 55. And the results are promising, where here 2 vaccine doses were given. And after the single dose, there was immune response within 14 days. And there was significant humoral and cellular immune response after 28 days. And this is just a single dose that was given. So most of the vaccine candidates right now in the market are all 2 vaccine doses. And as everybody would understand, getting 1 vaccine dose is tough. Getting 2 vaccine doses within a span of 15-20 days can be even tougher. Right. So this is a single shot vaccine, but they are right now doing additional doses in phase 2B of the trial, as well as they are starting phase 3. The phase 2B is generally directed towards the older people. So they have not divulged much. So looking at this, you can really understand that probably people who were older were not getting good immune response. That's the reason they are giving a second dose of the vaccine. And this was published in Lancet, and so all of this data have been peer reviewed. So that is one thing that people should remember, and people should understand, that you should not just go by what is being circulated around in WhatsApp, Facebook, or in news or in print media. We should be looking at peer reviewed journals to really understand whether a vaccine candidate is actually working or not. And the next is Cyanobac, where the phase 2 trial was completed with 600 people. Again, it happened only in a single province in China. Here, the vaccine was given into people with 18 to 59 years of age. It's a 2-dose vaccines where they can see neutralizing antibody titers. What does neutralizing antibody titers mean? That the antibody was able to kill the virus or neutralize the virus. So the new virus cannot infect any more cells, about to 90% of the individuals who got the vaccines after the 2 doses. And there is a phase 2B again with older people that are being conducted right now. So moving along, the AstraZeneca, the third of the vaccine candidate companies which are moving into phase 3. The phase 1, phase 2 study has been published, where 543 people were infecting. So they have 2 different lead candidates. I'm talking about the candidate which they infected with 543, that is moving into the phase 3 trial right now. So which was given in 5 sites in UK. So it was a single dose as well as a booster dose that were only given to only 10 individuals. And they saw that the booster booster dose after 28 days really gave a good immune response. And all the 10 participants who got the booster dose, all of them had neutralizing antibody titers. The people who got single dose, about 70 or 80% of the people got antibody titers. And this was published in Lancet last month. The fourth vaccine, which is heading to the phase 3, is the Moderna vaccine. The phase 1 trial has been completed in 2 sites in US. One was in Seattle and the other one was where I did my postdoc at Emory University. And here 18 to 55 year of age people were given the vaccine. There were 3 different doses that were tested. And as I said, Moderna is the mRNA-based vaccine, just for people to recapitulate. So here, all the 4 doses gave good humoral response after the 2 injections. There were neutralizing antibody response that were observed after the booster doses, but there was adverse side effects at the 250 microgram dose. So right now they're moving ahead with phase 3 with only 100 microgram dose, which gave a much better response, as well as there were no side effects with them. And currently they are also having phase 2 trial, which they have completed the enrollment. So they are performing the phase 2 and the phase 3 trial simultaneously. The phase 3 trial will have 30,000 people and the phase 2 trial has about 350 people. And I think, and this was published in New England Journal of Medicine last month also. And I think the last candidate that I would be talking about is the BioNTech Pfizer, where the phase 1 study was conducted with 45 participants, again with the lead candidates. There were other candidates that they tried and they're stuck with the candidate, which they are moving on to phase 2 and phase 3 right now. So they were given 2 doses. They were 21 days apart and they gave 3 different doses, 10, 30, and 100. This is again is an mRNA-based vaccine. The difference between the Moderna vaccine and the Pfizer vaccine is there were no side effects or adverse effects in this vaccine because the mRNA was encapsulated with lipid nanoparticles. And that is the reason it was much more safer and very little side effects, as well as this lipid nanoparticle, it helps this doesn't need to be frozen. It can be refrigerated. But for the Moderna, the mRNA was not encapsulated, so the vaccine has to be given, has to be shipped in the freezer, which again is a problem for developing countries, not just developing, for any country to have the vaccine in the freezer just before delivering. So this also provided humoral immune responses with neutralizing antibody titer that was detected at 21 days after the first dose and after 7 days after the booster dose. But again, this is a two-dose vaccines, and that is given. So as I said, so there are a lot of challenges with all these vaccine candidates that are starting phase 3 trial. So I have tried to highlight each of the challenges. But again, most of these vaccines are 2-dose vaccines, which is difficult. Considering that people need vaccine right now, and if you want to give 2 vaccines within a span of 1 month, then you have to manufacture twice the number of vaccines, which is difficult, as well as trying to bring somebody back into the clinic to give a vaccine after only 28 days is also difficult. And we still don't know, as I said, all of these studies have been done only for 2 months. So we still don't know how long this immune or the antibody response will last. So what is needed to cross this finish line of to have a vaccine in the market? I think it's a fine balance. The balance being first, the safety. That is the most important thing. You don't want to have a person get even sicker after getting the vaccine. And I can highlight one example that was actually happened for the case of Denvaxia, which is a dengue vaccine that was approved by FDA, and it has been manufactured by Sanofi after 20 years of research with $2 billion cost, where people were given, or kids were given this dengue vaccine in Philippines, and a few hundred people actually died because of providing the vaccine. The story why the people died is because of the antibody dependent enhancement. I don't think I have time to explain about why, but if somebody wants to read, there are a lot of articles on this. So you need to be very careful that the safety of whatever that you are administering is strong. So that's the first and the most important thing. The second is the efficacy. None of the vaccines right now that are on trial have determined efficacy, which means whether the vaccine will actually work. FDA last month gave a guidelines, at least for US, that the vaccine that they will actually approve should have at least 50% efficacy, which means people who have got the vaccine, about 50% of the people will be protected from COVID-19 compared to the people who have not got the vaccine. This 50% is not that high, but again, if you consider about flu vaccines that people get every year, this is not bad. The flu vaccine, the efficacy lies between 19% and 60%. So 19% is bad, right? So only 20% of the people are protected, but this is somewhere much higher. But if you consider the childhood vaccines like polio vaccine or MMR or any other vaccine, they have a 99% efficacy or 90%, or measles vaccines. They are really, really, really good. But at least for now, FDA has a efficacy limit set at 50. This is just for US. Other countries can have completely different efficacy guidelines. So it all depends upon how much risk that that country wants to take. But efficacy is something that is very important. And finally, the speed. Everybody is moving at a really high speed. We also need to, and that is important because as our principal mentioned, that about 17 million people have been infected throughout the world, and about 0.7 million people have died. So we need a vaccine sooner than later. So speed is important, but we have to have a fine balance between safety and efficacy. So all of these three things combined together, I think, can have a vaccine candidate which we can see in the near future. I don't think there will be a vaccine candidate this year, but we can expect to have a vaccine to be in the market maybe early next year or to the middle of next year. With that, I will stop and I will take any questions. This is where at Emory Vaccine Center where I used to work, and this is where the Moderna vaccine trial happened this year. And if there is any questions, please let me know.
Thank you very much, Dr. Bhomik, for your very informative and very beautiful presentation. There are a lot of questions, but I'll have to select out of that because of the time constraint. So my first question this is not from me actually, Devan Kormakar from YouTube link. He has asked, sir, can we afford herd immunity in India?
Sorry, but can you repeat the question? I just muted for a second.
Can we afford herd immunity in India?
I think that is a very good question. I think it is difficult to acquire herd immunity because, as I said, about 70% of the people needs to get infected. And with India being such a high populated country, and the hospitalization infrastructure is not very well equipped. I think there is, for every 1,000 people, there is one bed or something like that. So if people start to get really sick, it is very difficult to have a control on those people getting the right and proper treatment. So it is very difficult to have herd immunity in a country where there is a huge population with very limited infrastructure for hospitalization and clinical care.
Thank you very much. I'm moving to the next question. Which type of lymphocyte between T lymphocyte and B lymphocyte is the most appropriate to prevent COVID-19? This was asked by Dipayan Bandopadhyay from YouTube.
Yeah, I think both T and B cells play a role. Both of the T cell has its own unique role. As I mentioned, that T cell are the ones, especially the CD8 T cells, they're the killer T cells, which actually kills the infected cells. And most of these vaccine candidates that have been looking into, they are trying to have adjuvants and other or nanoparticles which actually boost the T cell response. But the primary focus of a vaccine is to generate a humoral response or B cell immediate immunity, which generates the antibodies which will actually neutralize. So that is the if you compare between the two, B cell immunity is the primary one, but you also need to have a strong, robust T cell immune response, which actually will give a much more better a vaccine candidate can become much better if it also has a good T cell response.
Thank you, Siddhartha. My last, because of the time constraint, I can only take this question as the last question.
Yeah,
so,
sure.
Dr. Sathi Saha, she asked, like HIV, is there any chance of transforming the surface protein of COVID-19? If so, how then the vaccine will be effective?
Till now, the people have been studying the mutations. I think I don't remember exactly, but I think there has been 50,000 or 60,000 gene sequences of COVID-19 that have been submitted, and there is a university where exactly close to where I work at Johns Hopkins, where they are analyzing each of the gene sequences that have been submitted for COVID-19, and till now there has been no evidence that there has been huge amount of mutations, especially in the spike protein where people are targeting. There has been some mutations that have happened, which has made the virus, or there is I can't say for sure, but there has been some evidence that the virus is getting fitter, as well as the virus is getting is being able to transmit faster. But the spike protein on which people are targeting to have the vaccines, that has not mutated. I think that's the most important key. So whatever you are targeting, if that is not mutating, there is a high chance that any vaccine will be efficacious. So I doubt the COVID-19 will not be as HIV.
Thank you very much, Dr. Bhomik. Once again, I would like to pay my sincere gratitude for your donation of time, of your precious time in our webinar. We hope to see you physically in our college in future. Thank you very much.
Thank you again, Dr. Sinha. But yeah, definitely. I think once we have a vaccine and everybody can travel freely, I'll definitely try to pay a visit to your college. Thank you.
Thank you. Thank you very much. Now, our next speaker is Dr. Mohammad Habibur Rahman. He will be introduced by Dr. Sunil Khan. He's a visiting professor of our department and ex-associate professor, SGB College, Bagati. Dr. Khan, please introduce.
Hello.
Please introduce Dr. Mohammad Habibur Rahman.
Good morning to everybody. May I audible? Hello.
Yes.
Okay.
Yes.
Yeah. Good morning to everybody. It is my immense pleasure, privilege, and great honor to welcome and introduce today's invited speaker in one-day international webinar, COVID-19 treatment and understanding on human immune system, immune response, a complex circuit for vaccine research. Today's second speaker is Dr. Mohammad Habibur Rahman, MD, PhD, who is a renowned nephrologist at All True Health System, Grand Forks, North Dakota, USA. He did his medical education from July 26 to June 2009 as a fellow of nephrology, SUNY, at the rate of Buffalo. July 2003 to June 2006 as hospitalist, internal medicine, Albany Medical College, Albany, New York. And September 1919 to June 2003 as a Ross University School of Medicine. He did his PhD in biochemistry, State University of New York at Buffalo. As a graduate PhD student at the University of Buffalo, he worked on a project to design a unique inhibitor for ribonuclease, which can inhibit and remove the most of the ribonuclease from aqueous media by one-step filtration. In 1996, State University of New York at Buffalo University. And he patented in 1996 his PhD work, which is very relevant in the context of the COVID-19 as because it is a RNA virus. Because this ribonuclease which can inhibit and remove most of the ribonuclease from the aqueous medium. In his credit, he has published 4 research papers, 8 research papers, national presented, and 6 abstracts. He participated in reviewing papers in peer-reviewed journals, actively involved in writing grant for funding National Institute of Health, Bethesda, USA, and National Cancer Research Institute. He has been working more than 10 years as a medical professional, and he will be very relevant. He will talk today on the topics of medical complications of COVID-19 patients, which we very eagerly want to listen in this pandemic situation. So over to Dr. Rahman for his presentation. Kindly.
Excuse me, sir. Please unmute yourself from your end.
Okay.
Okay, now clear. You are audible now. You are audible now, sir.
No, yes. I'm just. Okay. Hi. Good morning, everybody. I would like to thank. First, I would like to congratulate and thank Dr. Sangram Sinha for arranging this wonderful forum where we will be able to present our latest findings. And also, I would like to thank Principal Dr. Asim Kumar Samanta, who did the original introduction, and Coordinator Dr. Chatterjee, Dr. Sunil Khan, and I would like to thank Dr. Bablu Gain. And also, I would like to have my sincere and yes, who presented just before me, Dr. Siddhartha Vamik, and another speaker will be coming, Dr. Kaushik Rai Chodori. I'm going to talk about the current status of the COVID-19 disease which caused by the coronavirus. I'll go through the complications and the signs and symptoms, as well as the treatments. I am involved in active treatment in Altru Hospital for the COVID-19 patient, as I am a nephrologist, but all the COVID-19 people, most of them who are admitted to the hospital, they also develop the acute kidney injury and needed help. Some of them even gone to the extent that I needed to provide renal replacement therapy for them, including one form of the therapy that is called CRRT, that is continuous renal replacement therapy, which runs 24 hours without any stop. So after giving these complications, I would like to go through now and start the main topic of the discussion. Okay. What is that? Okay. I'd like to start with the signs and symptoms of this COVID-19 infection. COVID-19 actually is a coronavirus which caused this complication called COVID-19. And the symptoms, I start with anybody can have this, the one who is infected with COVID-19, they have the mild to severe symptoms. And the symptom severity actually varies from people to people and depending upon the pre-existing health condition. People who have pre-existing lung disease like COPD, asthma, or hypertension, even people with a chronic kidney disease and diabetes seem to have more severe complications. The symptom can appear between 2 to 14 days after the exposure to the virus. And most common symptoms are fever, chill, cough, shortness of breath, difficulty with breathing, headache, muscle ache, and one new actually phenomena which is very unique with the COVID-19 is the loss of smell and taste. They can have sinus congestion and some also develop severe diarrhea. Since this disease is a very evolving disease and we are still knowing this about the disease process of coronavirus, the symptoms, maybe other symptoms will be emerging in near future. I'll just focus on COVID-19 complications. About 1 in 6 patients will have complications. Some will have very severe life-threatening complications. And these life-threatening complications come from the cytokine release syndrome. And that happens when the infection triggers the immune system to release the protein that is called cytokines. And those cytokines are really affecting not only several organ system and even causing the end organ damage. COVID-19 complications, it starts with an acute respiratory failure where people have real difficulty with their breathing. And this symptom can appear within very short span of time after the person detected fever, chill. And as I said, that the severity depends upon the pre-existing condition of the person's health. Others is associated with a pneumonia, which is a secondary infection of the associated with the COVID-19 respiratory failure. Then acute respiratory distress syndrome, ARDS, that is also another type of presentation where you see the severely lungs are infiltrated, and that's why they call it a butterfly type of presentation in chest X-ray, where both lung is completely white out and there is no respiratory exchange system available and the lungs are completely filled. And those people are ventilated and sometimes they need 100% oxygen, but still they cannot keep their saturation at a target level. Sometimes those people can be placed in a prone position to have the oxygenation. And also sometimes they just now at our hospital, we have started to take the blood, pass through the oxygenator, and then send it back to the body so that the person can have enough oxygenation to the body. Because of these complications, it is so severe, and the severity, as I said, really depends on the people's pre-existing condition. But not only, there is a myth that the young people will not have it. I have seen in my hospital with 20 years old people who passed away, and it cannot be said that only the older people, but yes, older people and pre-existing condition people have the severity of the same symptom, but young adults who can also have the symptom. And then another one is the acute liver failure. That is also seen very commonly in the COVID patient who are severely ill and who needed ventilation support. And the most likely cause here is the disseminated intravascular coagulation, where the our own coagulation pathway do not work well, and the people starts to have severe bleeding. And in few cases, we have seen those people got into the acute fulminant liver failure, and eventually, because of the failure of the liver, those people's life just ended, and we could not do anything for those people. Other is cardiac injury, which is also the people who have a normal cardiac function prior to infection. And once they got infected and admitted, they can develop all of a sudden sudden onset of arrhythmia. And it is really very life-threatening for those people, even though they may have no previous coronary artery disease. Next is acute kidney injury, which I have mentioned. Acute kidney injury are all of a sudden, some persons kidney failure, and it can start with a mild injury to the severe injury. And since this, can you hear me? Okay. Since the infection can lead to septic shock type of picture, when there is a people with a septic shock, then the renal perfusion becomes very complicated. Those people develop acute tubular damage, and because of that, since the blood pressure is so low, they are hypotensive, and then we need to provide them with a pressure vasopressor support, and the normal dialysis system will not work for them. For those, then we have to do the continuous renal replacement therapy. We are able to see people when they recover the renal function, when they recover their illness. So depending upon the severity of the illness, some people at the end of the ventilator, when they get off the ventilator, still those people are on dialysis. Even though their blood pressure normalized, they are not on any vasopressor support to keep their blood pressure in a normal level. Whether those people will need continue in the dialysis in the near future, or they need to continue the dialysis, yet to be seen, because this infection is very new, and we are only 5 to 6 months into the pandemic, and we have seen several cases of people who had renal failure and the renal function not yet recovered. There are few people in our unit where with gone into the renal failure, they did not recover, and we are still considering them as an acute renal failure patient, and we are continuously providing them dialysis. Some people coming to the dialysis center 2 times a week, and some team come 3 times a week, depending upon their volume overload cycle and their improvement in the urine output. So at this point, we do not know whether we'll be able to set those people off the dialysis or not. still needs to be seen. But in an acute kidney injury situation, this can last for several months. Some people can have the renal recovery 3 months to 6 months. So we are hoping we'll be able to get those people off dialysis and they will be able to get back to their normal life. Other complications which we have seen very commonly of the people who got off the ventilation, their lung injury is so severe, those people are still needing oxygen, supplemental oxygen to do their day-to-day life. Even minor activity making them very dyspneic. They need 3-4 liters of oxygen even during the sleep. There are published report and also seen the another complication is called the COVID-19 fibrosis. That is, those people, their lung got damaged so severely that their oxygenation process is not normal anymore. Even with supplemental oxygen, they do not have adequate saturation. So there are cases reported now that those few of them have lung transplant to save their life. So this is a new disease, throwing some new light into our life. We do not know exactly how much the effect will be long lasting, starting from heart to kidney to lung. It's affecting multi-organ in our body. And people are dying. Yes, we have gone through many other epidemic, and we have not seen those. We are in this era which we are seeing, and people are really worried. And some people even I have seen do not want to come and get admitted to the hospital with a minor symptom. They say that if you have COVID in the hospital, then we may get it. But the healthcare providers are really taking good precautions not to transmit from one to other, but still some of them are very skeptical, and they just say, okay, I'm going to home and try myself. If I can get better, if not, then I'll come back. So saying that, I will say that it's a disease which is causing multi-organ failure. It is not only limited to the people who have previous health conditions or problem, but it is affecting people who have no previous health problem. And then another one is that the children, I have talked about the disseminated intravascular coagulation process, and in case of children, it's also causing multi-system inflammatory syndrome in children. If you have seen the news, then they have focused mostly in the New York, where they have more cases of the children admitted to the hospital, and they presented that. And some of those are really pathetic to see those young kids around 10 to 12 years to 18 years old. They have not only they are ventilated, but they have a severe rash in their body. They have the kind of a blue toe syndrome. Their toes in the fingers and in the hands turning blue, and their complaints. Most of them complain with a belly pain, vomiting, rash, and also this syndrome actually is now they are saying it is look like the old known syndrome, which is the toxic shock syndrome, and also known as Kawasaki syndrome. The children who have recovered, and they still have some effect on their memory. And it is not yet clear whether how much the recovery we are going to expect from those infection which affected very young lives. Those are not even less than 18 years old. Hope they are doing better, and the family is really devastated with this disease. So even though we are in a beginning phase of this disease process, but it is taking toll to not only to adults or older people, it is affecting the young children too. And the last one also, what I have seen. Also, we have seen with the people that is the rhabdomyolysis. Rhabdomyolysis is where you have a muscle breakdown, and the muscle breakdowns, once they have a muscle breakdown, they release the myoglobin. And the myoglobins are also very toxic to the kidney. They cause the pigment nephropathy. And in the pigment nephropathy depends upon the total CPK level, how much the total muscle damage happened. So, we have seen in our hospitals with the rhabdomyolysis cases where the total CPK level more than several hundred thousands. And we are actually able to save some of them, and few of them gone to in a state that even with the continuous renal replacement therapy, we could not save their life. So as we say that it is affecting multi-organ system, including, I did not see any cases that where what is the effect of the brain, what is the effect, what is the ultimate effect on the brain when those people got extubated after prolonged hospitalization. The people, once they got intubated, I have seen them, they stay in the ventilator support for 13, 14 days. And after 13, 14 days, when you have a hypoxic brain injury, we do not know how much those people's brain function or the memory will recover. Some of them I have seen, they are gone home, but even after 2 weeks of out of the ventilator, those people could not recall their name. Those people, some of them could not recall their date of birth. So still we are following those people, and maybe in the future, I'll be able to update how those people are doing who are ventilated for more than 3 weeks. So these are the complications which I have noticed in my care, providing care to the hospital. And I do not know how much more we'll be able to see. And I do not know how much it is going to have long-term effect on our neurological, renal, or cardiac complications in the future once those people go out to the go out of the hospital and go back to their home. Next. Yeah. So with this now, I'll just go give some just few lines about the testing process for the COVID-19. There are 2 types of testing. One is your viral test, where you do test the person who is infected, and viral. If the viral testing is positive, the person has the infection with the COVID. And second is the antibody test. Antibody test is just to see that the person developed the immune response and they have the antibody. That means these people were infected and or had past infection. So next is. Now I'll go over with a very little about the treatment procedure, because this COVID-19 is a very new disease to us. We don't have previous knowledge of this, and not many treatment procedures or medications are available. As Dr. Bhaumik already mentioned in his lecture, that we are just in the development stage of vaccine, and which is far away. We don't have any, we don't know when the vaccine will come. But there are few treatments what we are providing in the hospital, and which are FDA approved for the treatment of the severity of the COVID-19 patients who are admitted to the hospital. The people who have the mild symptom, most of them are recommended to stay home and stay in a quarantine state for 10 to 14 days. And if there is a fever, then take the medications, acetaminophen, Tylenol, or the NSAIDs, that is ibuprofen, naproxen, Aleve, Advil, those medications. But the problem with the NSAIDs that they are also very badly affects the kidney function. So even the CDC initially recommended not use of the NSAIDs, but later they recommended it. but it's still the mainstay of therapy. If someone has fever or chill, take Tylenol and or acetaminophen, and take NSAIDs, that is ibuprofen, when we need it to fight the breakthrough temperature or fever. So that's the specific for the people who have mild symptom. But for most severely complicated people, they are admitted to the hospital. Then either they are on a supplemental oxygen or they are in a assisted ventilation status. As I said, therapeutics are still in infancy and it is in investigative state. And some of them are approved by FDA. We are using them in the hospital. And those are, some of them is those, we have the anti-malarial drug and antiviral drug, but these are not new. These are old drugs, but we are using them. And the antibodies from people who recovered from the COVID-19 infection. And the first one is the convalescent plasma. In the convalescent plasma is the people who recover from the COVID-19 infection. And those antibodies are given in a transfusion form to the people who are severely sick. And the donor antibodies, they help to fight the illness and shortening the length of the hospital stay and reducing the severity of the disease. In USA on March 24, 2020, the FDA allowed the use of convalescent plasma for patients with serious or immediately life-threatening infection. So people who are infected, they can donate or they can give their plasma, but there are few criteria that needs to be met. That their antibody is enough. There is enough response from the virus that they have generated enough antibody. Then those antibodies are collected and then they are tested for HIV and also Hep C. And antibody, another thing is that once you collect antibody, there can be given to 1 to 3 people maximum. That much of the antibody they produce. And this is the first one to be given to the very severely sick people who are intubated. Let me just see 1 minute. And next one, I'll just go. This is the first one actually. So first one is the convalescent plasma. And then let's go to the second one. That is the second one is the antiviral drug. And those antiviral drug, they do not kill the virus, but instead they reduce the symptom and lessen the hospitalization. There are one antiviral drug which is currently used, and we have been using them in our hospital, is the remdesivir. This remdesivir, yeah. So remdesivir is given to the people, and it showed that it reduced the hospitalization or recovery by 33%. That is the people who received the remdesivir and the people who did not receive the remdesivir, their timeline around 11 days to 15 days. The people who received the remdesivir, they come off the vent or come off the oxygen, supplemental oxygen in 11 days, but versus the people who did not get it. And remdesivir is another, is that if it is given to a people who are intubated, that do not help. The people get benefited who are on supplemental oxygen, they recover better. So it is recommended not to give to the people who are intubated, but given to the people who are in supplemental oxygen. And it is given 6 to 5, it is given 5 to 10 days for IV. It's an IV drug. There is no oral formulation. So even for the minor symptom, there is no oral formulation of this drug that can be given to the people to recover. Only hope that they produce enough of this remdesivir to supply to different hospitals and can be given to the people who are still struggling with a breathing problem and having still want supplemental oxygen, whom cannot be weaned off the supplemental oxygen. There was a significant shortage, and in our hospital, we did not get it for quite some time, the remdesivir. But for now, the supply chain is good, and we are getting it, and we are giving to the people who need them, and who are only people who are getting benefited, who are on supplemental oxygen, but not to the people who are intubated. Intubated people is still getting that convalescent plasma. And how far the benefit is there, but people, the published work from the China that showed there is improvement, but there is no randomized control trial. So since the lack of the control trial is here, we do not know if some of them who got off the ventilator support is because of their own immune response improved or the convalescent plasma helped. But since it is helping, we are giving them, and we are hoping that it is helping them, because it had convalescent plasma, use of convalescent plasma is not new. It has been used in the past too. So that is the status of this two things, that is convalescent plasma and remdesivir. Now the third one is the dexamethasone. It is a corticosteroid, and it's an anti-inflammatory drug. And it is being given to treat and prevent the organ dysfunction and lung injury from inflammation. And this new study found that it also reduced the death by about 30% to the people on ventilation and 20% of the people who are not, who are on supplemental oxygen. And NIH recently recommended the use of this dexamethasone to the COVID-19 patient who are on mechanical ventilation. And if it is if it is given to the less severe people, infectious people, then it is harmful rather than useful, because they suppress the immune system. So it is only recommended to given to the people who are on supplemental oxygen or who are on a ventilated state. So for minor or for the mild disease, it is not recommended at all. So main therapy is the three-prong now. One, we are using convalescent plasma. Second is the remdesivir. And third is the dexamethasone. We are using all three of them depending upon the case status. People who are intubated and ventilated, they are getting dexamethasone and convalescent plasma. People who are on supplemental oxygen, and we are giving them dexamethasone, as well as we are giving them the remdesivir. So those combination of medications helped people to come off their ventilator state as well as supplemental oxygen. But as I said, there is no oral medications available yet, and people getting better and improving with those. Mostly these three are the mainstay of therapy at this point in USA: remdesivir, convalescent plasma, and dexamethasone. Other treatment I'll go over, but just like the one which is started, chloroquine and hydroxychloroquine. There's a chloroquine and hydroxychloroquine is not a new medications. They are on available for a long time, using for malaria and the lupus. And it started to use from the news that China has used them, and then they got benefited. But in USA, we use them, And there are many publications already in Lancet and also VA, and it is not helpful. It rather caused new cardiac arrhythmia and fatal death. In some institutions and some research organizations, they stopped the use, or some of the hospitals stopped the use when they found that death rate with the arrhythmia is much higher. So they did not use that medication. So hydroxychloroquine or chloroquine alone or in combination with azithromycin. Azithromycin had some anti-inflammatory drug. It's not an antiviral drug. It's a macrolide. It's a antibacterial medications. But because of the presence of the anti-inflammatory effect, researchers and also providers thought that it may help to improve the status of the health, because COVID-19 is an inflammatory, severely highly inflammatory process where it is causing the cytokine burst. But unfortunately, the published paper did not show any benefit of using chloroquine or hydroxychloroquine alone or in combination with azithromycin. And even, yes, another thing, the FDA actually revoked the use of chloroquine or hydroxychloroquine to the patients who are hospitalized. Then since people have tried multiple things, they also tried the monoclonal antibodies. Monoclonal antibodies are engineered antibodies, which we use very randomly and very frequently for the cancers. For cancer now, this is mainstay of therapy in cancer to use the monoclonal antibody. They are less toxic and showing really good promising sign for the people. And two of such candidate is tocilizumab and sarilizumab. Both have been used in a trial in different hospitals, but the outcome is not great. There is no significant benefit, and it is not being used anymore in anywhere in the United States. I have discussed about the azithromycin, and the last one people are using vitamin D. And vitamin D, actually, we know that the people with a low vitamin D are susceptible to upper respiratory tract infection. So people have been using vitamin D, and there is no really randomized trial to show that the vitamin D is efficacious and helpful to prevent the COVID-19. But since it is people with a low vitamin D are susceptible to upper respiratory tract infection, it is thought that it may be helpful and maybe at least preventive to COVID-19 infection. So people are using it, but I have not seen any randomized trial that I can say that it is helpful. But since it is a benign medication, it is not harmful, anyone can take it. And there are good sources in the food cycle, we can get it, fatty fish, tuna, mackerel, and salmon, milk, and also cereal fortified with vitamin D. So I can tell that, I mean, since it has no side effect. Of course, vitamin D toxicity is possible when you take too much dosage, more than 4,000 or 5,000 international unit a day. But if you use 400 or 500 international unit a day, it is not going to have any effect on you. So if without any side effects or without any significant problem, I would say take it if it is not going to get any problem. Just like omega-3, I mean, it is used, people use it from the fish oil, people use it to for coronary artery disease, improving HDL in our blood. So this is exactly kind of the same thing, that we can use it. It is not causing any health damage. It is not affecting any other behavioral aspect of our life. So we can take it. Next is a vaccine, but actually, my previous speaker already gave a detailed presentation on the vaccine and vaccine development. And I don't want to go much detail into it. And we don't, the thing is that now still it's in infancy. We do not know. And one thing is, I want to add, the people who are infected and recovered from the coronavirus, when their plasma was taken for the antibodies after the infection, and 3 to 4 months later the infection, the antibody level gone down, and in some cases, the antibody level is undetectable. So the question is, those people who are infected at one point and who recovered, and those people do not have the antibody present in their blood, they are again ready for infection. So my question is that if that is the status, then what will be the vaccine situation? Because the vaccine should protect, provide an immune response, and that will just like a measles, mumps, rubella, those vaccines are, once you take them, it's a lifelong, you don't get it. And they protect you 99% of the response, have the 99% of the response. So far, in what I have seen in the vaccine development and vaccine, people are still puzzled that whether we'll have an effective vaccine that will provide us long-term protection. That is a big challenge now. It is a big challenge to all scientific community. People are around the globe working on its development. The previous speaker already mentioned that there are more than 170 vaccine candidate online, and there are 27 vaccines currently on human trials. So the fate of the vaccine is uncertain, and we do not know when they will be coming, whether they will be available this year or by the end, or the beginning of the 2021. So best suggestion at this point is, since we do not have any oral medications as a prophylaxis that we can take, people started to take hydroxychloroquine as a prophylaxis. But I have seen data, I have seen people who are lupus and who are taking lifelong hydroxychloroquine, they got COVID-19 infection. So hydroxychloroquine is not a magic bullet that will that we can take it as a prophylactic medication to get, or that can prevent us from getting the COVID or the coronavirus infection. And since there is no other medication on horizon, which I have in my practice, I have not seen anyone available at this time that we can take orally, that to as a prophylaxis to prevent any COVID-19 or coronavirus infection. And only thing that is now proven and helping is the safe distancing, as it is mentioned, social distancing, face mask, hand hygiene, and staying away from big gatherings, stay home. That is the mainstay of prevention at this point. And I hope we should all follow this guideline. And as you are saying about in India, how we can do it, we are really have thickly populated country. And in that place, I think we have to officially mandate to the people to use the face mask. And even if it's social distancing is not possible, because the virus spreads through our respiratory droplets. If we put the face mask, that is providing a significant protection to the person who is next to me. Okay, so that will help to protect myself and protect the person who is next to me to from getting that infection. So there is one person, actually, it is not published in any paper, but there is one barber in New York who was positive for coronavirus. And he used the face mask and he did hair cutting for other people whom he asked them to put the face mask. And it was found that those 5 or 6 people whom he did the haircut, none of them got the virus. So the face mask, it works. It is not it is not just a theory, but it works. So my message to anybody, anywhere listening me, that please use the face mask, use hand hygiene, and practice social distancing. So that is the mainstay now. Vaccine is far away. There is no oral medications. And I have discussed the serious complications associated with the COVID-19. So in order for a safety of yourself and safety for others, that is the best message I have for everyone. Thank you.
Thank you very much, Dr. Rahman. I really acknowledge you for investing your precious time in spite of your busy schedule. You manage time for us. Thank you very much on behalf of the organizer and myself. There are many questions, but due to time constraint, I will be able to pick up on some selected questions. My first question to you that came from Dr. Shatabdi Ghosh from ISI from June. Dr. Ghosh asked why dexamethasone only acts on the patients with severe symptoms.
Okay, because the dexamethasone, when they are severely sick, our adrenal gland, which provides the stress hormone, is not working. So when the people are extremely sick, then if you give the dexamethasone, then that takes over and helps the stress related hormone. When it is absent, dexamethasone provides that support and provides the for the adrenal crisis. It do not happen in that case. So in order to prevent adrenal crisis in such a case, the dexamethasone is really, really important and helpful. So that's the message here, because if you are giving them, you don't want to suppress your own stress response. So as much as you can to have your own stress response, that is the best. So you don't want to suppress it. So dexamethasone is given to a severely sick people who cannot develop their own response to the stress.
Thank you.
That's the reason. Yeah.
Next question from Anirban Paul. He asked it from YouTube that if antibody test screening a person found positive, what does it mean that this person never gets infected by coronavirus again in future, or this person is immune resistant?
Yeah, so that's a good question. Actually, the thing is, we have seen, as I mentioned in my previous talk, in my talk, that people who are infected in the past and they have developed the antibody. That's what we are talking about, the herd immunity. In a herd immunity is that once you have the infection, then you are protected from the infection. That is, you will not get that infection again. But in a coronavirus, it's a really a virus of, very unpredictable path, an unpredictable working hypothesis. Here it is developing the antibodies, but the antibodies is disappearing within 3 months. They have tested that. So since the antibody is not there, if we have an antibody that protects against the new infection, that's when the herd immunity will work. Because if we develop the antibody, that means the people who have the antibody, they will never get that disease again. But here, the antibody is disappearing. So the person is ready for another cycle of infection if he come across with someone who is infected. So this is not, see, that's the problem. That's why I was talking about the vaccine generation, that the vaccine will how will you generate a vaccine that will give you the immunity for a long period of time if you cannot generate the antibody which will last longer? So that's the problem. So that is that if you are infected and since the antibody is disappearing within 3 months of your time, then after 3 months, it is there. It happened here also in US, that the people who are infected once, their antibody disappear in 3-4 months, and within 6 months time, those people got reinfected. So it is not protection. We can get reinfection again.
Sir, one more question. This is the last question. Dr. Sathi Saha from YouTube, he asked, all are talking about COVID-19 risk to diabetic people, but people who are already prone to infection, that is having infection-induced hemolytic anemia, how can they be safe in this pandemic situation?
Okay, so those actually, the diabetes actually is a disease of a systemic disease. It affects each and every organ, including eyes, our peripheral nervous system, and it's an inflammatory process. So people with, that's why the people who are diabetic, they are more prone to get that infection because of their own inflammatory, their highly inflammatory state. And the infection is affecting them more because of their own immune system. So that is the reason they are more affected by the COVID-19 than the other people. Also the people who are other immunologically suppressed, that the people who have a transplant, so upon organ transplant, those are immunosuppressed people, and they are more prone to get infection. The other group of people who are significantly at risk are the people with a previous lung disease. That is, those people have their already, when people are COPD or asthma, they are all chronically on a medication which are steroid based. And that is another inflammatory system. So people who are already in a state of inflammation, they are more prone to get the disease than the people who are not.
Thank you very much, sir. Dr. Habibur Rahman, thank you very much from the organizer and myself. Good night, sir.
Thank you. Thank you.
We enjoyed a lot.
Yes.
We have learned a lot from you. And I hope our audience are very satisfied and they are very happy with your presentation and elaboration and with your informative talk. Thank you very much once again.
Thank you. And yes, if you have opportunity, I can get it, and I'll give more information as the time progress. And if we have more data, since I am working in a hospital, if we have more data in which we see that if there are any other medications which are efficacious, and if I can update, I'll be happy to do that. And thank you for inviting me, and I will be available in future for presentations if I need to come, if I am be helpful for anything, and if I can provide any new information, I'll be happy to do that.
Thank you very much, sir. So kind of you. I hope we'll get your suggestion, and sometimes whenever our audience may contact you in future. Okay. Thank you very much, sir.
Thank you. Thank you. All right. Thanks.
Now, I'm moving to the next speaker, Dr. Kawasaki Chodori. He is basically immunologist. He's not working on COVID-19 right now, but he's a classical and applied immunologist. Without understanding of the basic immunology, many of here are students and research scholars. So I think this lecture will be very helpful to coordinate with the other lectures delivered by the speakers, Dr. Bhomik and Dr. Rahman. So with this, I would like to introduce our respected distinguished speaker, Dr. Kawasaki Chodori. He worked on his PhD jointly at the Indian Institute of Chemical Biology and the University of Lübeck in Germany, where he studied host pathogen interaction between the Leishmania donovani, the causative agent of Kala-Azar, and the human macrophage at a molecular level. Dr. Roy Chodori showed that Leishmania pathogens have a receptor that mimics the human chemokine receptor CCR5 and is chemo-attracted towards the macrophages. Macrophages in turn engulf the pathogen without inflammation. And the pathogen establishes itself in the macrophages. From there, Dr. Roy Chodori moved to Washington State University, St. Louis, where he developed techniques to identify point mutation using next-gen DNA sequencing. Dr. Roy Chodori later moved to the Cincinnati Children's Hospital at the University of Cincinnati in Ohio, USA. There he established the molecular mechanism of neuronal stem cells differentiation in early embryos. He is currently a faculty of pediatrics at the university and is course director for the master's program in biomedical research. A new generation MS program is entirely hands-on, laboratory-based, and students perform first-hand research on induced pluripotent stem cells, making CRISPR and transgenic mice and next-gen sequencing, among other things. Dr. Roy Chodori also teaches microbiology, epidemiology, and public health at Miami University, where he holds a concurrent visiting faculty position. He has published many research papers and research articles in peer-reviewed journals, and he is a very pioneer worker in chemokine receptor. When it was just evolving in the science arena, he started working at ISCV. With this little introduction, the CV is very, there are a lot of informations in the CV as well, but with the time constant, I would like to confine my introduction with these little words. I hope if the audience will understand, because of the time constant. Dr. Roy Chodori is waiting already. I think there is 12 o'clock in USA, so he will be delivering his speech. May I request Dr. Roy Chodori, please deliver your speech.
Thank you, Dr. Sinha, for that very kind introduction. Am I audible?
Yeah, absolutely.
Great. So, good morning to all in India, and good evening to my fellow participants here in the US, and a good day to everyone else around the world. So I would like to start by thanking all the students of Vivekananda College, Haripal, and all the other students who are there for giving up this beautiful Saturday morning to enlighten yourself on this very important topic. It is almost 1 o'clock at night here, so I'll try my best to hold your attention and not get you bored over there, because you are giving up your valuable time, so I hope you get something out of this. I want to first extend my thanks to Dr. Shamantu for kindly inviting me to talk to this August gathering here today. I also thank our speakers, Dr. Bhomik and Dr. Rahman. I've known Dr. Bhomik for a very, very long time, actually, for their seminal presentations today. I also extend my gratitude to Dr. Khan and Shri Bablu Kain and the governing body. আপনাদের অনেক ধন্যবাদ এখানে সাহায্য করার জন্য. Finally, I have to thank Dr. Sangram Sinha, my longtime friend, my ex-colleague, for organizing this wonderful symposium. And at that, without further ado, I would like to start talking about host pathogen interactions. I'm not sharing my screen yet, so it's not a malfunction. Before I share my screen, I want to introduce you to the concept of a constant and evolving war. A war that happens all the time between the host and a variety of different pathogens. And this has been raging for billions of years. This has been evolving for many, many years. And this is akin to a war insurgence. If you think of the Second World War, there were so many insurgencies where a small group gets in and tries to devastate, whatever country they're attacking, whether it was the Allied forces or the Axis forces. And we are seeing similar insurgencies these days around the world from various different groups who want to destabilize other places. As insurgents develop sophisticated weapons and they try to destroy a nation from its inside, at the same time, the nations, these armies, the police forces, and all the paramilitary forces, they are also constantly evolving their own tactics, their preventive tactics. They try to contain the current emergencies that are emerging, and they learn from that, and they try to try to prevent future insurgencies of that type. So something very, very similar occurs within our bodies, within our system, in the host pathogen interactions. In my talk, which I'll try to keep as concise as possible, I will introduce you to some of the fundamental concepts, some of the weapons that a lot of pathogens use, including our COVID-19, and some police and commando forces, that we have, our immune systems can deploy, and what happens when suddenly an unanticipated militant like the SARS-CoV-2 or COVID-19 gets into the system, and we really do not know what to do with it. Do we just let it get in and do a lot of damage? Or do we mount a response, and do we sometimes, go overboard, which does happen in real war situations as well? If you deploy an armed force, an army within a civilian society, what does happen? You get a lot of collateral damage. A lot of innocent civilians get killed. No war is good. I'm pretty against war of any kind, but inside our bodies, we have to fight this. And a lot of collateral damage does happen in a lot of cases, including in COVID. And sometimes the remedy can be worse, the cure can be worse than what caused the problem in the first place. So with this brief introduction, I'll share my screen and get to my first slide. I am going to start with the arsenal of weaponry that many pathogens have. Okay, please let me know, Dr. Sinha, can you guys see my screen? I am guessing that's a yes. All right. So.
Yes, sir.
Great, thank you. So I'll start with the huge arsenal that a pathogen comes with. So any pathogen is equipped with a lot of different weapons, and different pathogens have different sets of weapons. Some have many different ones, some focus on a few. This, I believe, is an Apache helicopter, but I might be wrong. I'm no expert in this. And we call these factors that a pathogen contains virulence factors. So what virulence factors does a pathogen have? It's important to understand. So I'll just go over some relatively common virulence factors. But what are virulence factors? Virulence factors are anything that assists a microorganism in causing a disease. Now, what you have to understand is, from the microorganism's point of view, the microorganism wants to survive. It has evolved. I mean, it does not really make conscious decisions like us, but it has evolved to survive. It needs to, for its survival, infect the host. And sometimes for that, it has to do significant damage to the host. Now, there are various types of virulence factors. Virulence factors are molecules or cell structures that allow attachment to the host cells. You heard Dr. Bhomik talk a fair bit about the spike proteins of the COVID-19 virus, SARS-CoV-2 virus, that help in attachment to host epithelial cells. That is critically important. Without that, the virus would never be able to enter. And if you can somehow inhibit that by one of a variety of methods that are being tried right now experimentally, you will be able to stop that infection from occurring right at the beginning, right at the first place. There are also methods that a lot of pathogens use to evade the host's immune system. So as hosts, we just don't let a pathogen come in and destroy us. We will mount a response, and I will talk a little more about some of the different responses we humans mount. Not just humans, all mammals do these things. Some of the responses we mount and how we try to contain them. But pathogens also are smart, and they're not smart in the way we are smart. They don't think, but they have evolved a lot of mechanisms by which a lot of them can evade host responses. And finally, a lot of these virulence factors allow damage to the host organism, either directly or indirectly. By directly, a lot of organisms can come in and do physical damage. A virus that replicates in significant numbers in cells can destroy the cell, and they can go to many other cells and destroy those cells, which itself is huge damage, but also an indirect damage in the way that it can elicit an immune response from the host, which can be very, very damaging to the host itself, not just the pathogen. So these are various things that the virulence factors do. Some of the common virulence factors, adhesion and attachment to host cells. Now, fimbria and capsules are basically bacterial things, not viral. But these are things that help these bacteria adhere to host cells. What do viruses do? Viruses have proteins on their surface, often spike proteins or other kinds of proteins that protrude out, and that allows specific attachment to host cells. You see this type of attachment. This picture is not of a virus that I show here. It's a bacterium, but you see these receptors down here, and you see the ones on the ligand molecules on the pathogen, invading pathogen. This is a lock and key type of docking. So it allows several things. One is, of course, to get infected, there needs to be docking, but also it allows the virus to choose exactly what cell it's going to infect. If the virus gets into the wrong cell, the cell is just going to kill the virus, right? The virus does not want to do that. So it has evolved to infect only a certain kind of cell, and more importantly, or equally importantly, only certain organisms. So the sequences, the protein sequences in these ligands that these pathogens carry are specific for certain organisms. Typically, a pathogen, a virus or a protozoan or a bacterium that often infects, let's say, a dog or a bat or a civet would not typically infect a human. Similarly, human pathogens by and large will not infect a cat. There are some specific examples where they will be transmitted, but most pathogens would not. The only case that can happen is if some mutations occur in this little receptor, and then the change in this receptor allows this pathogen to actually start docking into a new species. This is relatively rare. Now, mutations occur pretty randomly. It's not just that mutation has to occur, but a second thing needs to happen, which is that this thing needs to actually get somehow transferred into this new host. So these two species must come in close contact at some point. And just because it gets into the new host by this receptor does not necessarily mean the pathogen will be successful in the new host. That is actually often not the case. And what we know from a lot of other diseases, including Ebola and a variety of other things, we know that these organisms sometime, now, sorry, I have no idea how these things are happening on my screen, these squiggle marks. I'm not doing them, but anyways, and now they're going away. Excellent, thank you. So even if it gets transferred to the new host, it may not be able to survive for very long. And it's not just survival, even if, let's say there is a second mutation and it is able to infect and survive in this new host, will it be able to be transmitted from this new host to yet another host? So like, let's say I get something from a bat, an Ebola. We still are not completely sure what's the causative agent of Ebola, what's a vector for Ebola, but let's say it's a reservoir. It could be a bat. It has been suspected for a while in Western Africa. And if it gets transmitted to me, chances are I will not transmit it to another person. But under certain circumstances, that can also happen. So transmission happens. But even so, what happens is after a few cycles through human hosts, often these pathogens become less active. They become less virulent. And I haven't gone into the concepts of virulence, but they become less potent in infecting, and at some point it goes away. However, occasionally what happens is as a third or a fourth mutation gets in, and the pathogen becomes really, really stable, and it can very stably over many generations keep transmitting from one human to another. And that is when we have a very, very serious problem, like we have now with our COVID-19 virus. There are a lot of other virulence factors. One is resistance to engulfment by host cells. So there are host phagocytes, host immune cells that try to eat up the pathogens. That's one of the first responses our body cells do. Neutrophils are the first guys to go in there and fight it. And then there are macrophages, dendritic cells, so might come later on. But these things try to just, eat up the pathogen. And a lot of times they figure out ways to evade this getting eaten up. Some pathogens are really, really tricky, and they actually allow themselves to be eaten. One of these pathogens was one I worked with, Leishmania donovani, but HIV is also one of these things, which they are super smart pathogens where they ultimately end up going straight to the immune cell and they infect the immune cell. And it's something like, if there is a robber out there in the society and running around and everyone is looking for them, there's a police force looking for them, there are paramilitary forces or whatever. There are a lot of people looking for them, and it's hard for them to hide. But if somehow they can trick a police car in thinking that, hey, I'm one of you guys, can you let me in? And if he's sitting inside the police car, no matter how hard you look all around, you won't be able to find that. And some pathogens do that. They hijack the immune system and they sit right inside the immune system and destroy everything else, including the immune system. HIV is a classic example of doing that, where they start off, often M-tropic ones start off with macrophages and then move on to T cells. HIV ultimately moves on to T cells, and they wreak havoc on the host immune system. And then there are yet other pathogens that do more trickery, and this is where it gets a little more complicated. So pathogens often display molecules on their surface, which our immune system has learned how to read and then develop proper attack methods against those things. It's like, getting a specific stamp, a specific signature of a particular invading species. And all right, if I see these molecules, that means this is a really bad guy. And I want to attack this with all the force that I have, whatever police forces we have. So we know that signature. However, what some pathogens do, like trypanosomatids are classic examples, and Leishmania does that to some extent. They display a certain antigen, an immunodominant antigen on their surface, and the host starts developing a lot of immunity against them. Cell-mediated immunity, humoral immunity, tons and tons of antibodies. So now the host knows that this is the molecule to look for. Only this organism has this molecule. Let us make a huge amount of antibody and attack it. Great, it usually works. But these organisms have developed methods where after some time, they just completely downregulate that thing and they no longer display that antigen anymore. And now they're displaying a totally different antigen. Now viruses can't switch so easily. Viruses do the same thing sometimes, but based on mutations. They can mutate and their receptors can change structure. Other parasites can actually actively do that from their transcriptional machinery inside. And now they are starting to display a totally different antigen than they started with. And the body by now has made lots and lots of antibodies against something, but with that, it can no longer identify the pathogen. And after some time, what it does is then the body starts mounting another response against this new thing that it sees. By the time the body has enough antibodies against this thing has again changed its outside coat and has morphed into something totally different with another antigen. And trypanosoma can do this like hundreds of times, hundreds of different things. And each time you're going to this 4 to 6 weeks of massive antibody response. You're making IgMs, IgG, all of that. And in the end, Lakshmani also does that. And in the end, the patient ends up with hypergammaglobulinemia, which means you have tons and tons of antibodies. I mean, your blood is thick with antibodies. If you take like serum from these patients, you will see that it will like kind of solidify over time. We used to always have this problem when we were collecting patient sera. How do you keep it in a good condition without these changes occurring? And so that's another trick that many pathogens do. Finally, this very interesting concept of molecular mimicry, where the pathogen tries to mimic a certain host molecule. Now, they're pathogens, they're totally different species. They should not have any of our molecules, but they have some molecules that look very, very similar to something that we have. And they're trying to display this and say, look, I'm one of your guys, It's like a fake passport you're taking to another country. And yeah, I belong here, And a lot of times the host will get fooled by that. Not always. And this can work in various different ways. It can, sometimes it's related, the host will recognize that as a foreign pathogen, but it will start raising antibodies against something which is very similar to some host cells. And then those antibodies are going to go and attack the host cells, or even cell-mediated immunity. So it can cause autoimmunity, which can be a big problem. And sometimes it's just hijacking the host system. The host chemokine system is hijacked like that by HIV, which binds to the CCR5 receptor. During my time, I found a Leishmania system that is involved in mimicry. So that's another fascinating area in host pathogen relationships that many hosts use, including a lot of viruses use these things. And so these sorts of reports are just starting to come out. This was just published this July, where SARS-CoV-2 is apparently also involved in mimicry. But that paper was mostly describing autoimmune problems, but also other mimicry problems. All right, so what is this host system we're talking about? Let me get into this. So there are, and you heard both Dr. Bhomik and Dr. Raman talk about these two arms of our immune system, so innate immunity and adaptive immunity. Or they were talking about humoral immunity and cell-mediated immunity. So let's get into dabble into a little bit of the nitty-gritties, and I'll try to keep this as generalized as possible without getting technical. Let us think of this scenario where, we are getting infected by a virus, let's say the nCoV-14. But before we get into that, let's talk a little bit about innate defenses. So these are defenses that we just by our structure have. Physical barriers, like our skin. Our skin is a wonderful barrier, If you touch a doorknob that has COVID-19 virus on it, you will not get infected. If you touch your nose or your eyes or your mouth with that hand, yes, there's a good chance. But just by touching the doorknob, it's not going to enter through your skin. Our skin is a beautiful defense mechanism, These are tough cells, they're full of fibroblasts, and it's shedding all the time. There's a dry layer on the outside, and it's not easy to infect intact skin. Now, it's cut skin, it's different. I still don't think the COVID will be able to get in and infect very easily because we don't have the right receptors there. But who knows? We don't know a lot of things about the COVID-19 virus. But generally, skin is a great defense. And then we also have a lot of different chemical substances. Sebum, this oily substance that in this hot summer we often get. These can actually be protective in various ways. And then we have these innate immune cells, which are now seeing, once again, I'm talking about June and July reports in the literature on nCoV-2, the SARS-CoV-2, which where innate immune cells are playing a huge role. And after the innate immune responses are cells that are not, and it's cells and can be complement too, but mostly cells that are basically, they do not know this pathogen, but they know something foreign has come in. So they know self versus non-self. Our immune cells know all our self cells. All our self cells have a certain signature on them, on their MHC class 1 molecule. And these cells know what the self cells are, and they can always identify, or a lot of times they can identify a foreign entity. The moment that they see a foreign entity, they can just go and attack that. So there is that innate immunity. And then there's adaptive immunity. If innate immunity is not sufficient to take care of a pathogen, then we use a different mechanism where our system learns. It learns from what components does this pathogen have, and can we find something in our repertoire that will be effective. So the analogy here would be, you have an insurgent attack or a armed robber attack someplace, and they're shooting lots of people, and the local police are unable to take care of this person. They know some really bad people are there. They're doing havoc. They are not able to. They don't have the right weapons to neutralize this threat. What do? Of course, you call some, rapid action force and some paramilitary force who might try to bomb them or something. But what if even that does not work? Then you try and find out, do we have any specialists that know the tactics of this particular organization or these people? Can we find any signatures in them, and can we identify what they're using? And can we then deploy those very specialized forces? Can we adapt our forces to start dealing with that? And that is what adaptive immunity is. That is where the body learns from this molecule and then finds something that will be effective. And this is also pretty much deployed in case of our SARS-CoV-2 or COVID-19. And there are two arms of that: cell-mediated immunity and humoral immunity. As the name suggests, this one is mediated by cells, and this one is stuff that's secreted into the solutions, basically antibodies secreted by B cells. All right, so let us play out a scenario. What happens when a pathogen, let's say like the SARS-CoV-2 virus, enters our body? And let us say it enters through our nasal epithelium, and it infects the epithelial tissues, it docks inside you. So let us say it just gets in there. What happens next? And I have drawn some very preliminary primary PowerPoint shaped cartoons here, nothing fancy. And here you have a virus that is docked onto a host cell. And this is true for any pathogen pretty much, or most pathogens. They use this common mechanism. So the pathogen has docked onto the host cell. And what's it going to do? It's going to enter the host cell next, either by the help of the host cell itself or by some other mechanism. It enters the host cell, and then it becomes an intracellular pathogen. Once it becomes an intracellular pathogen, the host cell starts expressing certain molecules on the outside that distinguishes it. But the host cell realizes something is wrong. Most of our cells are capable of sensing that some foreign entity has got in, or something is seriously wrong. And our cells have the ability to call for help, to cry for help, send out beacons. It's very similar to, if a gathering of people or if a house has been invaded, we can phone call, we can shout from our windows or balconies and ask for help. And that's exactly, of course, the virus is going to replicate within the cell, but it sends out these stress molecules, these distress molecules. Often these are chemokines, chemoattractive molecules, and it starts sending many of them. And these are very specific. They're trying to call a certain kind of cell. Usually it's your local police they're calling. And the local police for our body system are neutrophils, neutrophil granulocytes. These are the largest bulk of our white blood cells, circulating white blood cells, are neutrophil granulocytes. They are the primary first responders to the scene of crime here. And the cells start secreting out these chemokines. And once they secrete out enough, what happens is this gradient forms that nearest to this infected cell, there are lots and lots of these chemokines. As you go out further, you see that this gradient gets thinner and thinner. And let's say we have a neutrophil, which is floating around, going along with the blood, and it senses some of these chemokines here. And it immediately knows some cell is in trouble. It starts following the chemokine gradient. It follows the chemokine gradient from, so these chemokines, it has receptors for the chemokines, and these, it follows the gradient of the chemokine, and the gradient leads it to the cell in distress, in our case, the infected cell. And so now it sees, all right, this cell is infected by viruses, and now viruses are coming out, budding out of this, and it's going to infect a lot of other cells. So the first thing the neutrophil is going to do is going to try and eat up these viruses. If it's a bacterium, it'll eat up the bacterium. Whatever pathogen, it's going to try and eat up the pathogen. Now, neutrophil response is also one of the very early responses during the COVID-19 virus infection. And the neutrophil eats up as many viruses as it can. And in a lot of cases, what happens is this solves the problem. You know, the neutrophil has come in, your local police have just come in, and they have neutralized the threat. They have taken care of it. They have arrested everyone or killed everyone and gone. And maybe one or two cells, a few cells died, all right, they will be replaced. Your problem is solved. This can happen, which is good news. This is what is supposed to happen. And we suffer some damage, not a lot of damage, but we recover relatively quickly. And this can happen. And the neutrophil is going to destroy all of that. But a lot of times, well, not a lot of times, sometimes the neutrophil gets overwhelmed because the threat is huge, or the pathogen that's invading has so many virulence factors in its arsenal that the neutrophil cannot handle it anymore. What is the poor neutrophil going to do over there? Well, the neutrophil can also send out radio signals, not radio signals, chemokine signals, of course, to ask for more powerful forces over there. And so the neutrophil sends out this chemokine gradient, and now a much bigger cell, the macrophage, comes in, and the macrophages, these guys are very, very good at eating up lots and lots of stuff. So they do a lot of phagocytosis. And the first thing that the macrophage will do is phagocytose and try to clear up all of these invading pathogens. Now, that's not the only thing a macrophage does. A macrophage do a lot of different things. It can also be a dendritic cell in some cases, so which can do this. But let's talk about the macrophage now. And the macrophages and a macrophage can engulf many, 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 many pathogens within itself and destroy all of them. And once again, in a lot of cases, this is going to, take care of the situation. Now your threat is neutralized. The body can heal back whatever cells were damaged. We can, replace those cells by making new cells. And all is nice and good. Many times that's going to happen. And well, sometimes the macrophage's efforts are not sufficient. There are some pathogens which can, outwit the macrophage or outcompete the macrophage. And the macrophages is outcompeted. It cannot fight against these many pathogens. So what does the macrophage do? The macrophage does a very interesting thing, and dendritic cells also do some things very similar. They try to neutralize as many of these as possible. They try to fight as many of these as possible. But when it sees that it cannot handle anymore, some of these macrophages capture a few of these guys, bad guys, and they sneak out of the scene. They are making a retreat, but they're not just running away. They have another mission later on, which I'll come to when I come to adaptive immunity. They are going to try and they're going to go throughout the body, millions and millions of cells. They are going to try and find a match, and they're going to go to every kind of cell, immune cell out there, and they're going to present this antigen to them and say, hey, do you have any weapons to try and neutralize this? Do you have anything that can be effective? And they're going to keep on presenting it to many, 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 many cells, mostly T cells. And until it finds one, and it will ultimately find some cell that gets a perfect match, and that cell says, oh yes, I have this perfect weapon that can completely destroy this thing. And then we'll make lots and lots of copies of that particular cell. And then those cells are going to come. So that's going to come on. So some of these guys have now sneaked out at this point. The infection at this point is pretty serious because your neutrophils have failed. Macrophages in their primary role as engulfing, they have failed. Some of them have other plans brewing up. What does the poor neutrophil do? And it could also be the macrophage. It sends out another kind of chemokine signal. Now these cells are desperate because now lots and lots of damage are happening, and even the immune cells are dying. So what does this desperate cell do? It calls one of the nastiest guys in its security system, the natural killer cell. And NK cells are very, very good at viruses and other pathogens. And one of the ways in which NK cells attack the pathogens is that they can shoot out these molecules called granzymes, which are lytic enzymes and other proteins, straight at the pathogen. And these granzymes, they are boom, boom, boom. They go and hit the pathogen, and they can kill the pathogens. Unfortunately, effective as the natural killer cells often are, they also do a lot of collateral damage. It's not just the pathogens that die from the granzymes. A lot of your host cells, even immune cells, macrophages and neutrophils can die completely, get pulverized, and a huge inflammation starts over there when the natural killer cells get crazy. So sometimes they're necessary, but the body waits a little bit before deploying these things often because they can also do a lot of damage to the body just as collateral damage. And you can may bring out the same analogies in times of a war in a city or someplace, where domestic forces might actually do a lot of harm to their own constituents they're protecting. So this part is your innate immunity. Now, as I said, the macrophage, it can sneak out and it processes the antigens from this from inside, and it purifies some of them and it presents it on its outside. It loads it on its MHC class II molecules. And then it goes to a lot of different T cells who have receptors, all kinds of receptors, but none of them are matching to what this guy has loaded, this macrophage has loaded on its surface. And it keeps doing that till it ultimately at some point finds a T cell that actually has the right match in the T cell receptor. And once there is this match, that T cell starts clonally proliferating, and it also starts secreting cytokines and interferons, and with all of these things, they go and attack the pathogen, and the pathogen would likely be neutralized. At the same time, a different arm of immunity gets triggered. B cells can directly sense these little pieces of the pathogen, the antigens, and there's also presentation that occurs. And there are a lot more nuances here that I'm not going into, because there is also B cell and T cell interactions that happen. There can be macrophage B cell interactions that happen. But the B cell senses and you find finally a B cell that has the right kind of antibody, that can make the right kind of antibody to neutralize these antigens. And it starts making lots and lots of these antibodies, and it secretes these antibodies into the bloodstream. The antibodies, they ultimately go to wherever the infection is, and many of the antibodies will neutralize the pathogen. What a vaccine does is, in a vaccine, it's like a pathogen. The body sees it like a pathogen, although it does not cause the disease. And you can have both cell-mediated immunity as well as humoral immunity, antibody immunity against that. And what we want to do is have initially at least antibodies against the pathogen, but for long-term immunity, we would like to have some memory cells and possibly some cell-mediated immunity component as well, which a good vaccine should elicit multiple mechanisms of protection. Okay, so this again is from a relatively recent picture. SARS-CoV-2 virus here is seen infecting epithelial cells, mucous cells. And then what happens? At day 1 of the infection, you have the virus infecting these cells. And very soon after, you get a variety of cellular responses. You have cytokine productions that are happening from a bunch of different cells. You get natural killer cells. Natural killer cells are very common way for viral infections. They're very, very good and effective in viral infections. And there are times when they can be effective. Now, the repertoire, as far as I'm not a medical doctor, but from what I've been hearing now in seminars and symposia, are that the variety you're getting of severity you're getting from patients is very, very huge, from completely asymptomatic to very, very severely affected individuals. And I wouldn't be surprised to learn that the virus gets controlled at various stages for different people. So there are natural killer cells, and then of course macrophages come into the play, where macrophages can directly neutralize these viruses, but also they can act as antigen presenting cells, and they can actually elicit cell-mediated immunity with T cells. But at the same time, B cells get activated and they start making antibodies. So plasma cells, plasma B cells will secrete antibodies. And ultimately, you have a response. Is that necessarily good? To some extent, yes. These early responses are definitely important to keep your virus in check. But what happens, as I have been saying, is that sometimes our immune system goes into hyperdrive. They see a threat that is much bigger than the threat really is, and they can mount a very, very robust response, which is good to get rid of the pathogen, but also does a lot of collateral damage to our host system. And that's what has been happening in this disease a lot in the more serious cases. And this is again from a very recent review that came out in Pharmaceuticals. Once again, this is from July 2020, where you see all of the different, this is summarized from a bunch of papers that came out in the last couple of months, where you see all of these different immune responses simultaneously happening in the COVID-19 cases, ultimately going one of two ways, immunological silence and immune suppression, or and systemic inflammatory response, which ultimately ends up in this cytokine storm, and is not good for the host because there are just so many responses being mounted that are a lot of collateral damages occurring. But as you can see, pretty much all components of our immune system gets activated with this disease. So what do we do about it? I think one group of scientists are trying to see if they can find substances that can enhance the beneficial factors of immune enhancement. We definitely need certain amount of immune enhancement to clear the virus, to go and kill the bad guys. You can't really kill a virus, it's a particle, but to inactivate the virus. But also at the same time, reduce the pro-inflammatory factors that lead to hyperinflammation and the cytokine storm, and can we selectively downregulate certain cytokines and certain responses that are more destructive to us than it's doing good by getting rid of the pathogen. So a huge amount of research right now is going on into immunomodulators. Until we have a vaccine, we have to try out different methods. I mean, we know some things that don't work, some things that work, and our previous speaker actually talked in a lot of depth about that. Of course, our first speaker gave us hope about a vaccine. Until we have that, we have to try and figure out, I believe, what are the other things that can modulate our immune system and give us some temporary level of protection if it is possible. And Dr. Rahman talked about vitamin D as a possibility. There are a lot of other things I'm seeing in the literature. I actually am not really in a position to say or judge which ones work and which ones don't. I have not worked with any of these personally. All right, if we have a few more minutes, it's already pretty late. Okay, I won't take a lot of your extra time. One of the things that I do is I teach a fair bit, and I teach epidemiology. I'm not getting into the epi triangle that I start my college lectures with, but I'm just going to talk about the chain of transmission model, where you have pathogen in a reservoir, and I'll come in a moment at what a reservoir is. And there is some way from this reservoir, the pathogen gets transmitted to a host, to us. We are the hosts in this particular case, and it comes in through a portal of entry. And this basically, and another way is the epi triangle. If you can break any of these links, you have successfully stopped transmission of the disease. And what are the reservoirs? There could be a lot of different natural reservoirs out there. Humans are great reservoirs of a lot of diseases, especially all the anthropogenic transmitted diseases. HIV, AIDS, but also SARS. Many diseases transmit from one human to another. Not all, like malaria would not typically, unless there's a blood transfusion, you don't shake hands with a malaria infected person and get malaria. You need an intermediate host in between, an intermediate zoonotic host, a vector in between to transfer it from one person to another. But a lot of times humans are reservoirs. In this particular case, humans are great reservoirs. Animals can be reservoirs, and we know that this virus is capable of infecting certain other animals. We do not know how good it is for transmitting that the virus from that animal back to humans, whether that is a big problem for us, because we are talking about vaccinating and eliminating the virus, and we can vaccinate some people or a lot of people maybe. But what if the virus lives on in some animal around us, and if the vaccine is not a lifelong, there's so many unknowns over here. If the vaccine just gives you an annual protection, like the influenza vaccine, just 1 year, and then your immunity goes down, and would you get back the thing? Would you have to keep getting vaccinated over and over again? There are lots of unknowns from what I understand about this disease. And there can be also non-living reservoirs, food, water, soil. Thankfully, as far as we know, this particular virus is not foodborne or waterborne, or does not do great in soil. But these are the reservoirs. There's also have to be transmission, movement of the disease from source to the host. And we by now know fairly well how this is transmitted. Direct contact transmission is often used by a lot of different pathogens, and of course, in very close contact, many pathogens can get transmitted from one person to another. But one major route that we understand this pathogen is using is direct droplet transmission. When a person, this is a high-speed photography of a person sneezing. When a person coughs or sneezes, all of these droplets are emitted. It happens so fast, we usually don't see all these droplets, and there isn't enough contrast, but lots of droplets are going out. Usually they, just settle down, fall down in less than a meter or in about a meter's distance. And so that's why we are trying to maintain 1 meter or 6 feet distance from each other to prevent this type of transmission. Masks help, as we have learned very clearly. And these are very things that now every child knows, not just my epidemiology students. Direct vertical transmission, we don't know much about what happens with the COVID-19, but this is mother to child transmission. We know certain diseases can get transmitted that way. Vector-borne transmission, again, we do not believe this is a vector-borne disease. So an insect or an arthropod or that is not involved as far as we know, but we don't know for sure, of course, but we don't think it is. Indirect vehicle transmission, once again, things that where the pathogen can get attached to water, food, etc., non-living transmissions. And then formites, things that a patient touches and then another person touches, a bed sheet, a blanket, and this could be one of the ways by which we know that the virus survives on these surfaces for a fair bit of time. So some percentage of patients, it's entirely possible, are getting this infection from these routes. But one thing that I was talking about earlier, this droplet mediated transmission that we all know about, that you have to be like, more than 6 feet apart, and then you're kind of safe, you're wearing a mask. So when you sneeze or when you cough, all these droplets are going out in the air, and the big pieces, the big droplets, they do fall very quickly. But there are lots of also tiny droplets, and there is virus in those as well. And often what happens is because they're so tiny, they float for a little while in the air. And before they can fall to the ground or whatever surface is around, they dry up. And what are you left with? You are left with just the pathogen, the virus in this particular case. But if it's a bacterial disease, just the bacteria floating around in air, you have an aerosol of the pathogen. And these things, we call them droplet nuclei. The droplet nuclei can stay airborne for many, many, many hours, and they can drift long distances with the air. It can get even worse when you are in a centrally air-conditioned place, whether internal air is circulating, let's say, and the filters are not all that good. You need very, very fine pore filters, something like a HEPA filter, to get rid of viruses. Really, droplets can be, a surgical mask will prevent droplets from getting out, but for the viruses that are dry, you need very fine pore filters. So that can be another problem. And then, of course, we need to know the portals of entry and exit. Usually undamaged skin is not a good way of entry for most pathogens. Some certain fungi can actually support themselves on dry skin, but most things cannot. Mucous membranes is a huge source, especially the respiratory mucosa, which we know is a very, very rich source of getting this particular virus, but also gastrointestinal mucosa and genitourinary tracts. They all have mucous membranes, and different pathogens use these modes for getting inside. All right, so how do we control the disease? We have to break the chain of transmission at one of these steps at least. For example, if you can treat ill people with antimicrobial drugs, which we do not have so far for the COVID-19, or reduce the number of reservoirs. Here in this case, we have human reservoirs, so you can't really reduce the number of humans. You can keep them away from other humans. You can decontaminate the vehicles, reduce the number of susceptible hosts by vaccination, and all of these things are things we are going to try in the future, I'm sure, once we have a vaccine, once we have a drug. But for now, all we can do is, avoid this aerosol transmission as far as possible and stay more than 1 meter away. And of course, the clinicians will be able to speak to this a lot better than I can. And this is common knowledge for everyone. And with that, I would like to end my presentation. Thank you, everyone, for keeping your attention. And I hope I did not bore you too much. I will be happy to take any questions that are there. So I am handing this back to you, Dr. Sinha.
Thank you very much, Kaushik. You have invested so much of time, even at 1 p.m. in your US residence. And thank you for your elaborate and very beautiful presentation. There are many questions, but I would like to take few of them because of the time constraint. The first question came from can intake of zinc, vitamin B3, C reduces the chance of getting COVID-19? If you have any idea in this regard.
Unfortunately, I do not have any information on that, and so I would be very hesitant in giving out any health advice here because A, I'm not a clinician, and B, I do not want to give out any kind of advice that may not be appropriate. So I will refrain from answering that question. Sorry.
The next question is from Dipayan Bandopadhyay. Is opsonin antibody related with macrophages?
So, well, yes, opsonins yes, it can be. So what happens is there is another method that I did not talk about here for a variety of reasons. But one of the things that happens is one of the crosstalks between the humoral immune system and the cell-mediated immune system, especially macrophages, as you pointed out, is that these antibodies can coat a pathogen and then can make these pathogens very, very easy for macrophages to identify. And once a macrophage sees an opsonized pathogen, it will trigger. So macrophages can take up cells in two different ways. One is like there are dead cells in our system. Our cells normally die, and they're replaced by new cells. And one of the things macrophages do is they go and scavenge these dead cells and they eat them out. But then it does not elicit a significant immune response. It does not start an inflammation over there, because otherwise we would always have inflammation throughout our bodies. Normally it should not. It's a silent uptake and it's just clearing out. It's doing one of its roles. But if it sees something with antibodies on top of it, it's going to engulf that and it is going to start an immune response and inflammatory response. So yes, there is that connection. Yeah.
Thank you, Dr. Chodori, for answering these questions. Once again, I would like to pay my gratitude to you for your kind presentation and donation of time in our webinar to make it successful. Thank you very much. Good night.
Thank you, Dr. Sinha. Thank you very much.
Thank you very much
for having me. Thanks.
Next, we are going to the plenary session. This will be chaired by our Honorable President of GB, Sri Bablu Gayan. So please, Devbroto, please transfer this to Sri Gayan if he is present here.
Yes, yes. I think he's not present here right now.
Okay. Can you find him?
He's not present right now. Hello?
Hello.
So he's not present here right now.
Okay. So in this plenary session, I would like to ask the experience and how the webinar, how did you feel about the webinar? So I would like to ask Dr. Chatterjee, if you are there, you can please interact. Hello.
Hello.
আপনি তাহলে একটু বলুন।
Yes. Carry on. Please carry on. Sir, please unmute yourself. Okay.
Please transfer the line to the president. He's in the webinar. Okay. Dr. Chatterjee, I'll request. I'm request to please. Our president sir is with us, so let him chair this session. Then I'll want to take some points from you also.
Okay, Bablu sir, please unmute yourself.
Yes, yes.
Okay.
So,
Yes, yes, you are audible.
Is there
ধন্যবাদ জানাই
হ্যাঁ
হরিপাল বিবেকানন্দ মহাবিদ্যালয়ের বটানি বিভাগের এইচওডি ডক্টর সংগ্রাম সিনহাকে এত সুন্দরভাবে বর্তমান করোনার আবহে চিকিৎসা পদ্ধতি সহ নানান বিষয়ে অভিজ্ঞ ডাক্তারবাবুদের দ্বারা এই সেমিনারের আয়োজন করার জন্য ওনাকে আমরা আন্তরিকভাবে ধন্যবাদ জানাচ্ছি এবং সাথে সাথে যারা ছিলেন আজকে ডাক্তার সিদ্ধার্থ ভৌমিক তিনি সুন্দরভাবে করোনা বিষয়টা কি এবং এ ব্যাপারে বিস্তারিতভাবে তিনি বলেছেন এবং ডাক্তার মোহাম্মদ হবিবুর রহমান তিনিও সুন্দর বক্তব্য রেখেছেন। সর্বশেষে ডাক্তার কৌশিক রায়চৌধুরী যেভাবে আমাদেরকে করোনার সম্পর্কে অবহিত করলেন, সচেতন করলেন এবং এর দ্বারা আমাদের গোটা সমাজ উপকৃত হবে বলে আমি মনে করি। আমাদের যারা এর সঙ্গে তারাও এ ব্যাপারে একমত হবেন এবং আমাদের হরিপাল কলেজের ছাত্রছাত্রীরাও অনেকেই এই সেমিনারে অংশ নিয়েছিলেন, তাদেরকেও আমরা ধন্যবাদ জানাই আমাদের কোঅর্ডিনেটর পার্থ চ্যাটার্জি সাহেবকে ধন্যবাদ, তিনি প্রথম থেকে এখানে হাজির ছিলেন। আর আমাদের তো সংগ্রামবাবু তো ছিলেনই এবং আমাদের প্রিন্সিপাল আমাদের অসীম কুমার সামন্ত তিনিও উপস্থিত আছেন এবং আমাদের গভর্নিং বডির মেম্বার হিসেবে প্রথম থেকেই হাজির আছেন আমাদের স্বরূপ মিত্র, ডক্টর করবী মান্না, ডক্টর আমাদের অভিজিৎ বাবু আছেন। সকলকেই আমি আমার পক্ষ থেকে ধন্যবাদ জানাচ্ছি এই সুন্দর অ্যারেঞ্জমেন্ট করার জন্য। ধন্যবাদ।
Thank you very much, sir. Now I would like to request Dr. Chatterjee, please share your experience regarding this webinar. Dr. Pathu Chatterjee, please.
Hello. Am I audible?
Yeah.
Yes, yes, you are audible.
Well, it was a very nice experience, starting from the early morning to hear to three experts from various fields, from vaccine development, from hospital angle, and also from the theoretical and applied aspects from Dr. Raichudhuri in the last. So I really like the session very much, and everybody has got a good lot of information from this webinar. And we hope that we'll get some vaccine as suggested by early in the Dr. Bhomik, that we can get some vaccine early next year so that we can get some relief from this pandemic that is going on throughout the world. So I once again thank the speakers on behalf of the IQAC Vivekananda Mahavidyalaya of being present in this webinar. Actually, it's late night, I think, there in USA. But we are in a fresh mood in the morning. So you might be very, having some tea also. So it is very nice to have you here all. Thank you very much to everybody for presenting and for participating in this webinar. And I thank Dr. Sangram Singha for arranging such a nice webinar. And also I thank the president of the governing body, Sri Bablugan and other members of the governing body present here and make it a grand success. Thank you very much.
Thank you, sir. Now I would like to request Dr. Abhijit Pakira, if he is there in the webinar, and can Devabruto can transfer this line to Dr. Abhijit Pakira? He is an honorable, respected GB member of our college.
Sure. Please put on your video. Sir, he is not present here.
So, আমি প্রেসিডেন্ট স্যারকে বলছি যে আর কেউ কি যদি কিছু বলার থাকে আপনি একটু বলে দিন, তাহলে দেবব্রত, প্লিজ টেল ইট টু আনমিউট।
Sir, please mute yourself from your end.
Huh?
Sir, please, Bablu sir, please unmute yourself.
সরল মিত্র আছে, দেখুন সরল মিত্র আছে।
Please transfer the line to Mr. সরল মিত্র।
সরল মিত্র।
Sharuk Mitra, please put on your video.
Hello.
Yes.
Video to start hocche na.
Okay, please accept the request.
Yeah,
okay,
now
now
you can see.
Hello.
Yes, you are audible, sir.
You are audible and visible.
Yes. Good morning to all honorable sir, principal sir, president sir, and madams, respected of facilities member of Vivekananda Mahavidyalaya, respected governing body members, and all designation present here. I think all of you organize a international webinar on COVID-19 treatment of understanding of human immunity response, a complex critiques of vaccine research organized by Department of Botany, Oripal Vivekananda Mahavidyalaya, collaborating with IQAC. In nutshell, I can say that I listen all the excellence present here very carefully, and I pray to the alumni that we can get right to COVID-19 parametric as soon as possible. Thank you so much, all of you. Thank you.
Thank you very much. Now, is there anybody who want to share the experience? President sir, is there anybody who want to share their experience?
No.
Okay, then we will switch over to the last part. Now, I would like to request Dr. Sunil Khan to please transfer the line to Dr. Sunil Khan.
Yes. Sir, please put on your video. Yes. Sir, please unmute yourself. Okay. Sir, please unmute yourself.
Yes.
Okay. Now.
Hello.
Yes.
Yes, you are audible, sir.
শুনতে পাচ্ছেন তো? ঠিক আছে। গুড আফটারনুন। শোনা যাচ্ছে ঠিক দেবব্রত?
Yes, sir. You are audible.
Okay. আর ভিডিও ঠিক আসছে? Is okay. Now it is okay. আচ্ছা। Good afternoon. It is my pleasure and privilege to introduce our convener, Dr. Sangram Sinha, who is a head of the department Botany, and we are one head of the department Botany, and we the organizing team are congratulating him for a nice arranging of webinar. Now, due to some time constraint, we have changed this program. We should have introduced Dr. Sinha earlier, but due to international time management, we change it to now. Dr. Sangram Sinha is a head of the department and assistant professor of Vivekananda Haripal Vivekananda College, and Dr. Sinha has received his PhD in the field of environmental microbiology from the Department of Botany, University of Colony in the year 2008. Dr. Sinha joined as assistant professor in the Department of Botany, Vivekananda Mahavidyalaya. He has been teaching UG course in botany for last 10 years and more than 10 years, and teaching postgraduate course in animal physiology as a visiting professor in the PG Department of Animal Physiology, Serampur College, Serampur. His core area of research is microbial bioremediation of heavy metal and arsenic from the soil and water. His research work is mainly focused on biomining of heavy metals, resistant and arsenic resistant bacteria for bioremediation. আচ্ছা এই অডিওটা কি স্ক্রিন শেয়ার হচ্ছে দেবব্রত?
হ্যাঁ ভালোই বলছি
স্যার, ইউ আর ভেরি মাচ অডিবল স্যার।
না স্ক্রিনটা শেয়ার হচ্ছে, স্ক্রিনে তো তোমাকে দেখতে পাচ্ছি।
হ্যাঁ আমি বললাম অভিজিৎ বাবু নেই।
আচ্ছা ওকে
শেয়ার করছেন?
না ওকে আচ্ছা, heavy metals and arsenic from soil and water by the bacteria. His research work is mainly focused on biomining of heavy metal resistant and arsenic resistant bacteria for bioremediation of heavy metals. Arsenic from wastewater, agriculture field, mine trailing, etc., and formulation of eco-friendly biofertilizer. He has received major research project from the UGC and Department of Biotechnology, Government of India. He has organized national international seminar in his department. He has been recognized as an independent research supervisor from the Department of Microbiology, University of Bardhaman. Presently, he is guiding 4 doctoral students. He has published many research articles and author of book chapters. He has been selected as a participant in the international training workshop on virus-free seed potato multiplication and disease integrated control techniques organized by the Virus-Free Seedling Research Institute, Heilongjiang Academy of Agricultural Science in Harbin, China. That's all. And I congratulate on behalf of the organizing committee, all the members and participants and all the dignitaries, speakers present. They were nice, beautiful webinar, very informative and very pertinent for this time of pandemic. Thank you all. ঠিক আছে, হ্যালো, আমার হয়ে গেছে।
ভোট অফ থ্যাঙ্কস এর জন্য আমাকে নামটা রেকমেন্ড করে দিন।
আচ্ছা, now I'm on behalf of the organizing committee requesting Dr. Sangram Sinha to present vote of thanks of this webinar. Now over to Dr. Sangram Sinha.
Thank you very much, sir. Respected principal, sir, Dr. Ashwin Kumar Samanto, Dr. Partha Chatterjee, IQAC coordinator, and Honorable President, Governing Body, Sri Bablu Gain, my distinguished resource persons, my esteemed colleagues, research scholars, students, and other dignified participants. It has been our pleasure to host this international webinar at Vivekananda Mahavidyalaya, Haripal, Hooghly. I'm thankful to the principal and President GB and IQAC coordinator, Dr. Partho Chatterjee, for giving me this opportunity to organize this seminar. Now, this is truly not a seminar, we call it webinar because seminar is not possible because of this COVID-19 pandemic. So in this kind of pandemic situation, these webinars, organizing webinars is a method to disseminate the knowledge among the students, among other research persons, and other common people to get to know about the treatment and strategies. How should they follow up the safety lines? And I also now would like to give focus on the topics that delivered by our distinguished speakers. Firstly, I'm thankful to Dr. Vomik for his beautiful presentation regarding the vaccine research. How is it progressing and what is the future of the vaccine research? And next, it was a beautiful deliberation from Dr. Habibur Rahman, who talk about the patient complications and what kind of treatment is now being used in the US and what are the drugs in different complicated conditions. So we enriched from his lecture. And lastly but not the least, Dr. Kaushik Roy Chodori, who is happened to be my very good and close friend, he delivered his lecture regarding the basic immunology, so that the students will be able to link up with the earlier speakers who who delivered the lecture on the vaccine research and the patient complication. In this way, with the collaboration from IQAC, with the collaboration from our respected principal sir and the President GB and other dignitaries and resource person, dear participants, my well-wishers, with all your sincere effort, collective effort, the webinar has ended up with a thumping success. Thank you very much for your kind patience from the morning. Thank you very much. Now with this, we're going to disconnect. Have a nice day.